University of South Carolina, and Dr. Christina Bassett, I don't know how to pronounce her last name, I'm sorry, from Dublin University College. So they're going to be moderating today's first beautiful session, and hopefully we have a great last day of PISA. Thanks for coming.
Yeah. Thank you, Sarah, for that introduction. Um, Me and Christina didn't decide who goes first, but I don't mind. What do you
You
think?
can go. You go ahead.
Okay, so it's my pleasure to introduce to you McKenna Grosses, who was doing research at the Medical University of South Carolina, and she hails from Elon University herself. And she'll be talking today about Alzheimer's disease and high-fat diet. So good luck.
Thank you so much. Oh, let's see. Okay, so Alzheimer's disease is a progressive brain disorder and the most common form of dementia, and is characterized by symptoms such as memory loss, confusion, and cognitive decline. Over 7 million Americans are currently living with Alzheimer's disease, and by 2050, this number is projected to rise to nearly 13 million. The lifetime risk for Alzheimer's at age 45 is 1 in 5 for women and 1 in 10 for men. Healthcare and long-term care costs for people living with dementia are projected to reach 384 billion in 2025 and nearly 1 trillion by 2050. This really highlights the significant healthcare burden to individuals living with this disease and highlights the need for more research. It's not entirely known what causes Alzheimer's disease, but its progression is linked to abnormal buildups of proteins from amyloid plaques and tau tangles. And this causes neurons to stop functioning, lose connections with other neurons, and die. And currently, there's no cure for this disease. Obesity is a significant risk factor for Alzheimer's disease, and a westernized high-fat diet has been known to cause obesity. Large-scale studies have shown that individuals with a body mass index of over 30 have a 90% increased risk of developing Alzheimer's disease compared to those with a normal BMI. Furthermore, obesity is associated with chronic inflammation, which is also proposed as a key driver for Alzheimer's disease and other neurodegenerative disorders. Previous studies have used APP/PS1 transgenic mice, which have mutant strains of amyloid precursor protein and presenilin-1, effectively giving them Alzheimer's disease, and a high-fat diet has been shown to increase Alzheimer's pathology in the brain. And most studies on Alzheimer's disease in both humans and mice have been very brain-centric, but nobody's ever looked at the stomach. For a little bit of context, the stomach and the brain actually have a direct connection through the vagus nerve, which has the highest innervation in the stomach outside of the central nervous system, which positions the stomach as a key organ and potentially showing that Alzheimer's disease might not be as brain-centric as we thought. A little bit of background on the stomach. The epithelium consists of these long gland-like structures, with each section being responsible for a different function. So the pit of the stomach has mucus pit cells, which produce mucus to protect the epithelium. The isthmus is known as the proliferative zone with progenitor cells. The neck of the stomach has mucus neck cells and parietal cells, which produce mucus and acid, respectively. And the base of the gland has chief cells, which secrete enzymes. When the healthy glands of the stomach succumb to damage, they become metaplastic and the architecture of the gland changes. We get an increase in mucus pit cells, a decrease in parietal cells, increase in tough cells, and increase in metaplastic cells. Typically, this is good. It comes in and helps heal the stomach and returns to the normal gland structure when it's done. However, we're seeing with the high-fat diet, there is gastric metaplasia, which we think could be releasing cytokines and increasing pathology in the brain of these Alzheimer's mice. So we had the Alzheimer's mice on a normal diet for 12 weeks, which we expected to find normal stomach glands, and a high-fat diet for 12 weeks, which we expected to find metaplastic glands. So first we looked at an H&E stain to observe the architecture of the stomach and found that there was first gland elongation signifying inflammation, along with additional mucus secreting cells in the high-fat diet. And you can see in the pink in the control, there's parietal cells, which we noticed there to be a loss of in the high-fat diet. So to further explore this, we did immunofluorescent staining, where we similarly observed a loss of parietal cells with the high-fat diet. This next stain's just a epithelial boundary marker, and then this last one stained for fibroblasts, which are known to be remodeled in metaplasia, which you can see here in this overlay. And after quantifying it, we did find a significant decrease in the parietal cells in the high-fat diet Alzheimer's mice. Next, we looked at MIS1, which stains chief cells. These chief cells are reprogrammed into metaplastic cells in metaplasia, and you can observe them in the control diet-fed mice, but not the high-fat diet mice. And we found significant decreases in chief cells in the high-fat diet. CD44 variant 9 is an established metaplasia marker. GS2 marks the mucus typically found in the neck of the stomach, which you can see in the top panel, but it's spread more throughout in the high-fat diet. And aquaportin 5 is another metaplasia marker. So in this overlay, you can see the glands that are metaplastic. So we counted the ones that were positive for all three of these stains and quantified the glands that we determined to be metaplastic and found significant increases in metaplastic glands in the high-fat diet Alzheimer's mice. High proliferation is another characteristic of metaplasia, and we found increases in metaplasia in the high-fat diet mice as well. And we also know that tough cells are increased in the high-fat diet metaplasia mice, which we observed here as well, and found significant increases. It's also important to note that these tough cells in the high-fat diet had long protrusions as to being more contained like they are in the control diet. And this is especially interesting in relation to Alzheimer's disease because tough cells are known to interact directly with the enteric nervous system, and these long neuropod-like protrusions can actually connect with neurons and help signal to the brain. So next, we revisited the H&E stains and found there to be immune infiltration in the high-fat diet-fed Alzheimer's mice. So that led us to wonder if maybe immune cells were what was driving the metaplasia. High fats are known to cause gastric damage, and when the gastric epithelium is damaged, this secretes alarmin IL-33. So we thought to look at IL-33 and found significant increases in the high-fat diet. And we next wanted to know which immune cells had receptors for IL-33 so that so these would signify that they're being recruited by the IL-33, and we found that mast cells have the highest receptors for them. So then we stained for mast cells. And in the high-fat diet, you can see there's an increase in the mast cells. We also did another mast cell stain for good measure with tryptase, and again saw similar results that mast cells were increased in the high-fat diet. Next, we looked at macrophages. These cells are known to be recruited by mast cells, and previous literature has shown that they have been known to drive metaplasia in other studies. So we looked at those and found that they were also increased in the high-fat diet. So next, we wanted to model this in vitro. So we turned to gastric organoids. But these organoids typically lose parietal cells, which we've established as a pretty important cell type when it comes to metaplasia. So to remedy this, we used immortalized stomach mesenchymal cells with the gastric organoids. And as you can see with this immunostaining with parietal cells in purple, we were able to maintain all the cell types in the gastric organoids. Next, we cultured these organoids with either a BSA control or palmitic acid, which is the main component in a westernized high-fat diet. And we saw that the palmitic acid-treated organoids were bigger than the control ones, and this could be signifying high proliferation, which is consistent with our in vivo models. That then led us to wonder if mitochondrial damage could potentially be causing the metaplasia, and parietal cells, we think, have high levels of mitochondria. So we stained for mitochondria with TOM20 and parietal cells. And you can see that where the mitochondria stain positive is also where parietal cells stain positive. But in this overlay, it's important to note that the mitochondria in the high-fat diet seem to be a little bit misshapen, which could be signifying mitochondrial damage. So to look at mitochondrial function, we put gastric organoids in a receiver, which measures mitochondrial function through the flux of oxygen output. And interestingly, we found that the palmitic acid-treated organoids had higher mitochondrial function, but we think that this could be triggering mitochondrial exhaustion down the road, which then could lead to the death of these parietal cells. So we know that mitochondrial damage causes reactive oxygen species, which also causes DNA damage. And interestingly, DNA damage is found accumulated in the brains of Alzheimer's patients. So that's what we looked at next. And in our high-fat diet mice in the bottom corner there, you can see that there's more spots for DNA damage and they're also brighter, signifying that there's increased DNA damage in the high-fat diet. Next, we wanted to look at proteins that are associated with Alzheimer's disease in the brain, and these are going to be alpha-synuclein and tau proteins that form aggregates in the brains of Alzheimer's patients. And we were looking to see if maybe these were happening in the stomach as well as the brain. And we found that there was increased tau proteins as well as increased alpha-synuclein proteins in the high-fat diet transgenic mice. And furthermore, in the overlay, you can see in the high-fat diet, these two proteins are actually aggregated together a lot more than they are in the control diet. So overall, our working model is that high-fat diets are causing mitochondrial damage, leading to inflammatory metaplasia, causing the release of cytokines, which could be impacting pathology in the brain, as well as these Alzheimer's disease-related proteins could potentially be forming in the stomach and traveling through the blood-brain barrier, also impacting pathology in the brain. Thank you very much.
Wow, that was really, really impressive, McKenna. While people are thinking of their questions to put in the chat or to scream out, I'll ask mine if that's okay. And so you very clearly show that there's this connection between the gut and the brain in Alzheimer's, and that it seems to be going from the gut to the brain. I was curious if there's any indication that the opposite direction may be happening, whereas maybe Alzheimer's patients have gut phenotypes. I'm curious if you've heard of anything like that.
Yeah, I'm actually not sure, but with the vagus nerve being there and them being so connected, I wouldn't be surprised. I think the tough cells also might be an indicator of that since they interact so much with the enteric nervous system.
Really interesting. Does anyone else have a question?
Hi, McKenna. Fantastic talk. I was actually texting it, like writing it in the chat just in case we ran out of time. But I really like that you're studying kind of the relationship between obesity and Alzheimer's. I don't know of many labs that have that ability to do so. We have an obesity and hypertension model because we study the cardiac, but I believe that you only used high-fat diet in your model. Is that correct? Just to induce the obesity side, not the hypertension? Have
Yes.
you been able to look in either your organoid models or at a just cellular basis, the lipid uptake ability of the cells and whether or not that's affected? Do you know if lipid uptake is affected in the brain during obesity as well?
Yeah, we actually haven't looked at that, but that's definitely something we could consider for the future. Thank you.
Awesome.
Any last minute questions? Well, thank you so much, McKenna, for your wonderful presentation. If anyone thinks of more questions, feel free to put them in the chat there, and she can answer those. I'm going to hand off our next introduction to my co-moderator, Christina.
Yeah. Okay, so now we move to the next speaker. We have the pleasure to have here giving a bit about her research, Cheryl Eka from the South Carolina Medical University, and she will talk about how a high-fat diet affects the pro-inflammatory needs and has an impact in developing gastric metaplasia.
Thank you so much for the introduction. Do you think high-fat foods are a problem only for your heart? Well, they also affect your stomach. Join me to find out how. Unhealthy diets are common, and the consumption of high-fat diets is associated with poor health outcomes and increased rate of obesity. Unfortunately, obesity is a worldwide problem, and the rates of obesity have tripled worldwide since 1975. And it is particularly alarming in the United States. Data from CDC indicates that in the United States in 1989, the obesity rate was less than 15% in all the states. 5 years later, 4 US states had rates of 16 to 25%, and less than 4 years later, half the states had an obesity rate up to 29%. And 20 years later, more than half the states have an obesity rate between 35 to 45%. Obesity can lead to many serious health conditions, including cardiovascular disease, type 2 diabetes, breathing problems, metabolic dysfunction associated fatty liver disease, chronic liver inflammation, and cancer. WHO recommends no more than 30% of your total energy intake from fats. A single burger has 21% fat, and a single slice of pizza has 15% fat. The effects of high-fat diet have been widely studied in mice, where mice are either fed with regular chow diet or high-fat diet, and the mice fed with high-fat diet gained weight to become obese. And it was found that these obese mice have altered gut microbiota, intestinal inflammation, loss of the mucus barrier, glucose intolerance, and increased risk of colon cancer. But what happens to the stomach? This hasn't been explored that well, and our lab studies the stomach. The stomach has three parts: the fundus, the corpus, and the antrum. Similar to humans, mice also have three parts: that's the forestomach, corpus, and antrum. The forestomach is more like the human esophagus with squamous epithelium, and we'll focus on the corpus region as it recapitulates the human stomach better than the other parts. Our stomach is lined by gastric glands that are composed of different kinds of cells. On top of the gastric gland is the pit, which is lined by mucus pit cells, and it produces mucus to protect the epithelium, followed by the isthmus, which has the progenitor cells, and it's also known as the proliferative zone. Next is the neck, which has the mucous neck cells and parietal cells. Parietal cells secrete acid that can activate the digestive enzymes, and then we have the mucous neck cells, which produce the mucus to protect the epithelium. Followed by the base, which has the chief cells. These are digestive enzyme secreting cells. We also have these red stop cells that are scattered throughout the gland, and they have chemosensory and immune function. We also have enteroendocrine cells that secrete histamine and hormones. Damage to these glands can lead to metaplasia, and it is characterized by increase in mucus pit cells, decrease in parietal cells, increase in tough cells, and increase in metaplastic cells. Sometimes these metaplastic glands can go back to their normal state after damage, but chronic inflammation most often leads to cancer. And the route to gastric cancer is a long multi-stage process. The death of acid-secreting parietal cells often leads to pyloric metaplasia, and which is characterized by increase in mucus-producing cells in the pit region known as foveolar hyperplasia, and at the base of the gland, there's an increase in proliferative mucus-producing metaplastic cells. Persistent inflammation during pyloric metaplasia may cause progression to incomplete intestinal metaplasia, which is characterized by the appearance of intestinal goblet cells. Pyloric or incomplete intestinal metaplasia may transition to complete intestinal metaplasia, which is characterized by the increase in intestinal cells like goblet cells, absorptive cells, and Paneth cells. The expansion of the disorganized growth in incomplete intestinal metaplasia may give rise to dysplasia, which is promoted by the stromal influence, such as fibroblasts. Dysplasia may then progress to gastric cancer. And gastric cancer is the fourth deadliest cancer in the world. And the risk factors for gastric cancer is H. pylori infection, dietary habits, ethnicity and geography, obesity, gender, tobacco use, and family history. And over the years, there has been a rapid increase in diets rich in fats in America, and this could be a contributing factor as we discussed before. There are several murine models for gastric metaplasia, such as the Helicobacter infection models, chemical models, ulcer models, and these are known to study gastric metaplasia. Our next question was, how does high-fat diet contribute to metaplasia? And we can study this by comparing our high-fat diet model with the established metaplasia models. This brings us to our preliminary data. Here we took black 6 mice and randomly assigned them into two groups. And we fed them with either regular chow diet, which has 10% fat, or high-fat diet, which has 54% fat. And all of them have same sugar levels. And we collected their stomachs and processed their tissues and immunostained them to look at the different cell types. I would like to thank Katrina Robinson's lab for providing us with the mice and collaborating with us. This wouldn't have been possible without that. And here we can look at the mice that were on high-fat diet. They gained weight to become obese. And here's an H&E stained control gastric tissue, that is the ones fed with regular chow diet. And we can see that they have mucus pit cells in the pit region. We have parietal cells. We have a lot of parietal cells in our control. We have the chief cells at the base of the gland. But when we look at the high-fat diet gastric tissue, there is a stark difference. The architecture is altered. We have elongated glands. Here we can see the expansion of mucus pit cells. We see reduced parietal cells and increased mucus cells. We also see that there's an increase in the infiltration of immune cells. All of this indicates that high-fat diet alters the gastric architecture. We wanted to further ask, how does high-fat diet impact gastric cells? So from previous studies on metaplasia, we will look at the gastric cell alteration with immunofluorescence staining. To look at parietal cells, we stain them with HK-ADPase, which is only present in parietal cells, and we see with high-fat diet, there's a significant decrease in parietal cells. And Claudin 18 is decreased. And we also can see this in the merged image. Upon quantification, we can see there's a significant decrease in parietal cells in our high-fat diet mice. And aquaporin 5, CD44 variant 9 are established metaplasia markers. And here's an example from literature to show the expression of these established metaplasia markers in gastric tissue. Aquaporin 5, CD44 variant 9, and GS2. Aquaporin 5 is a water transporter and is specifically present in the deep antral glands of the mucus cells and not in the corpus glands. GS2 is a marker of mucus and it's present in the neck region in the healthy glands, but not in the base of the glands. And CD44 variant 9 is a surface cell metaplasia marker and it's not present in the healthy corpus glands. Only in metaplasia, we can see the expression of aquaporin 5, GS2, at the base of the glands, along with CD44 variant 9 in the bottom panels. And now we look at these specific metaplasia markers in our high-fat diet tissue. And we see that with high-fat diet, there's a significant increase in the metaplasia marker CD44 variant 9. And Ki67, which is a proliferative cell marker, is also significantly increased. And you can appreciate the presence of both CD44 variant 9 and Ki67 staining in our mice on high-fat diet in these merged images. This is interesting because most of our proliferative cells, as I mentioned before, is in the neck region, in the proliferative zone, and that's the isthmus region in the normal glands. And these studies show that the metaplasia in the chief cells can transdifferentiate into proliferative metaplastic cells. Now we stain for aquaporin 5, another metaplasia marker, and we see a significant increase in mucus-producing metaplastic cells that have aquaporin 5. We also see that the GS2 is expressed at the base of the glands in these high-fat diet tissue, all indicative of metaplasia. And here is a merged image. Immune cells are known to play a crucial role in driving metaplasia. Previous studies have identified the damaged tissue produce alarmin IL-33 that recruit immune cells that can secrete IL-13 cytokine to drive metaplasia. This has been shown in several studies. Our control mice and high-fat diet mice express IL-33 in the pit cells. And we see that IL-33 expression is significantly increased in our high-fat diet mice. And this is a beta-catenin is an epithelial cell marker, and you can see that the IL-33 expression in pink in the high-fat diet is increased in the pit region, the lamina propria, and the base of the glands. And I think you can appreciate it here in the merged image. And IL-33 alarmin is known to recruit a variety of immune cells. And to look at the cells that express this IL-33 receptor, we use CellX gene that shows human single-cell data, and we utilized it to identify the cells that express the IL-33 receptor. IL-33 receptor is ST2 or IL-1, RL1. And we found that mast cells express the highest amount of IL-33 receptor. Then we stained for toluidine blue, which is a mast cell marker, and we saw very few purple mast cells in our control. But when we looked at our high-fat diet tissue, we see a lot of these purple mast cells. We also immunostained to look at mast cells with the help of tryptase, and we can see that in our high-fat diet tissue, there is a significant increase in tryptase-positive mast cells. And several studies have shown that mast cells can recruit ILC2s. Metaplasia models showed that induced metaplasia have heightened ILC2s and are important for metaplasia. So we next looked at ILC2s in our control and high-fat diet mice, and ILC2s marked by GATA3, which is pink here, is significantly increased in our high-fat diet mice. Next, we looked at macrophages, and studies have shown that macrophages are essential for metaplasia progression. And we see that in our high-fat diet mice, we have a significant increase in macrophages in the lamina propria and the base of the glands. And it's in the pink. And as I mentioned before, these immune cells would produce IL-13 that drive metaplasia, and it was found in a study that mast cells are the greatest contributors to IL-13. And IL-13 can increase tough cells and intestinal organoids, and studies have also shown that tough cells increase in metaplasia. So to look at tough cells, we stain for DCLK1, and we see in our high-fat diet mice, we have a significant increase in DCLK1 positive tough cells, and phospho-ERM1 also marks parietal cells and the tough region of the tough cells. And we see a decrease in parietal cells. And here's a merged image, and we can see that there's a significant increase in tough cells. And here is the quantification. Next, we wanted to see if fats alone can induce this effect. To address this question, we turn to gastric organoids, which are generated from the corpus glands and have all the cell types of the gastric epithelium. We treated these organoids with palmitic acid, the main component of high-fat diet, and DMSO as a vehicle control. We found that the palmitic acid could increase the size of our organoids, which suggests that these cells are highly proliferative. These findings mirror our in vivo findings that a high-fat diet stimulates proliferation. And we also stained for Ki67, a proliferation marker, and we see that palmitic acid-treated gastric organoids have a lot of Ki67 positive cells. And we have also developed some human gastric organoids from healthy person, and we'll be repeating our palmitic acid organoid experiments in here. Until now, we have seen that 25 weeks of high-fat diet decreases causes metaplasia, which is characterized by decrease in parietal cells, increase in metaplastic cells, increase in mast cells, ILC2s, and increase in macrophages and tough cells. And we hypothesize that the high-fat diet damages the gastric epithelium, and gastric epithelial cells produce IL-33 that recruit mast cells and other immune cells like ILC2s. And these immune cells produce IL-13 that drive metaplasia. And this leads to tough cell hyperplasia, chronic inflammation, and metaplasia. Thank you.
Thank you very much for your nice talk. Unfortunately, I think we're a bit too tight in time, so if there's any question in the chat, you can further discuss about how this research is happening. So now we move to the next speaker that Selene will introduce.
Yeah, so it's my pleasure to introduce to you Doug Terry. And so he's going to be talking to you today from Emory about how the ATPase activity can regulate intestinal restitution. And so we're very excited to hear from you. And so whenever you're ready, feel free to get started.
Great, thank you. Yeah, my name's Doug Terry. Thank you for giving me an opportunity to speak today.
I would like to interrupt you real quick. We see your presenter mode. So.
Oh no, okay. Let me see and show. It did not show up like that when we tried it earlier.
Yeah, when you were testing it, it was perfect.
Is it still presenter mode for y'all?
Yes. Oh wait.
Yeah, if it is presenter view, but if you go back and end the show, you can also click on play from start. And should you need your notes, I believe you can just do right click and that should be it.
Right click. How about that?
Or maybe not.
Certainly would prefer to have my notes. Let's see.
Currently, it looks good.
And now it's back to presenter.
Try clicking Use Slideshow on the top left.
Top left, use slideshow. Yeah, back to that same thing. Weird. Okay. Let's see.
I would try unsharing and resharing. It's possible. You click the the PowerPoint instead of the screen.
Oh, you're probably right. Yeah. Okay. Let's see. No success. Is it good now or no?
No. Hit exit one more
I
time.
think when you are sharing, you can choose instead of sharing your screen, share just the PowerPoint, like the slides. There's an option when you are sharing the screen.
So when I'm in,
While we're
then what
figuring
were you saying?
something out, oh, go ahead.
Sorry, what were you saying? Ah, it's okay.
I think what Fran was just saying was that if you end the show and then hit on the share screen, you should be able to select what view you want to display. So you might want to try that out. It might just take another second, but don't worry. We're not discounting your time.
Let's see. Let's see. Desktop. It only has the one application window.
So when you are in Zoom and you share your screen, you have always several options like the your whole screen or just like one of the like all the software you have open, all the programs. So you can choose
Yes.
specifically just PowerPoint instead of the whole screen. Does it make sense?
Yeah, so
You
I've
have that option?
selected, yeah, and I've selected the application here. Um,
Okay.
yeah. Is that, yeah. Um, but still just giving me this. Yeah. Okay.
Instead, let me try one thing. Exit for a second. Go to the bottom right and hit the presentation button. Because that always works for me.
Oh, for when I'm in sharing. Okay.
Yeah.
Let's see, go down there, share.
So it's on the bottom right of your screen. It looks like a little, I don't know, projector screen. Yes.
Okay. Continue to share. Yeah, and that also doesn't give me my notes. Okay. Um, well, I guess we'll do it without the notes then. This should be fun. Play from Cool. Okay. Well, yeah, we'll just go ahead and go. Yeah, so my name is Doug Terry. I'm a postdoctoral fellow at Emory University in the Robinson Lab. Yeah, we'll keep going. So in the Robinson Lab, we use complementary approaches in Drosophila, mice, and humans to study epithelial repair programs. And in particular, I'm interested in studying epithelial repair in the intestine. So, Drosophila is a good organism for studying highly conserved functions across metazoans. And one of those necessary functions is for a digestive tract that's able to absorb nutrients while protecting against a harmful, potentially harmful environment. And in particular, we're interested in we study the midgut of the fly, and in particular, the posterior midgut of the fly, which corresponds to the small intestine. And at a cellular level, the midgut of the fly is composed of three major cell types in a pseudostratified epithelium, which is much simpler than in mammals, but it's composed of three major cell types: progenitor intestinal stem cells, or ISCs, enterocytes, absorptive enterocytes, ECs, and enteroendocrine cells. Let me go and grab my pointer if I can. Yeah, great. Um, So some previous research from the lab looking at lectins that I don't have time to get into today found a potential role for lysosomes in regulating intestinal cell homeostasis in response to injury. And this isn't particularly surprising given the known roles of lysosomes in mediating endocytosis and phagocytosis, as well as a key role in autophagy. And we wanted to further study lysosomes. Lysosomes are highly acidified, and this acidification is largely produced by vacuolar ATPase proton pump, which pumps protons into the lysosomes. And so in our experiments going forward, we look at manipulating this vacuolar ATPase proton pump and the effect on oxidative stress in the fly. So we do this by feeding our flies an oxidative stressor that, as you might imagine, leads either paraquat or hydrogen peroxide, which, as you might imagine, leads to injury to the intestine and eventual death of the fly. So after we feed our flies these oxidative stressors, we can analyze the survival and perform molecular analyses. And we did this in either wild type flies or flies that had knockdown of this VATPase proton pump, and then studied survival. So knowing what we knew about lysosomes, that lysosomes are generally needed to help regulate the cell in response to oxidative stress, we were expecting that knockdown of these vacuolar ATPase proton pumps would sensitize the flies to oxidative stress. However, surprisingly, when we actually performed our whole animal knockdowns of VATPase subunits, we found that knockdown of these subunits was protective in our model. So I'm not going to get too far into the weeds on Drosophila genetics, but what I'm showing you here is in the green is a tubulin Gal4 drive, which is capable of driving knockdown throughout the entire fly. So in the green is our control, where we're not actually driving anything. And then I'm showing you knockdown of several subunits of the vacuolar ATPase proton pump. And when we knock down this proton pump, we see increased survival of the fly. But that was with the whole animal knockdown, so we wanted to see if that effect would localize to the intestine. So we used an intestinal specific driver to knock down our vacuolar ATPase subunits. And we found when we do this in intestinal stem cells, we don't see a significant protective effect of the ATPase subunit knockdown. But when we do it specifically in enterocytes using this MyO1A Gal4 driver, we see this strong protective effect. So going forward, we're going to focus on enterocytes, and everything I'm showing you is in enterocytes unless I say differently. So we want to look at a lysotracker, which stains acidified organelles, particularly the lysosome. And as expected and shown by previous studies, when we give our flies an oxidative stressor, we see an increase in these lysotracker staining organelles. And as you might expect, when we knock down a subunit of the vacuolar ATPase, in this case VHA44, which is an ortholog of V1 subunit C, we see a decrease in acidified organelles even following this oxidative stressor. So we next wanted to test if, try to identify what was causing this protective effect upon knockdown of our vacuolar ATPase subunits. So in Drosophila, death is often preceded by loss of intestine barrier integrity, and that either in whether that's death by aging or toxicity. And so there's this fairly blunt, simple assay that you can do in flies called the Smurf assay, where you feed flies a blue food dye and look for spread of that dye throughout the fly. So in healthy flies, that dye should be constricted to the digestive tract, but in flies that have barrier function degradation, you get spread of that dye into the hemolymph and throughout the body of the fly, resulting in this Smurf phenotype. So following oxidative stress, we have a high proportion of our flies show this Smurf barrier dysfunction phenotype. And when we knock down VHA44, specifically in the enterocytes, we see a suppression of that effect. Coinciding with this, we looked at death caspase 1 staining, a marker of apoptosis in Drosophila, following 24 hours of oxidative injury, and found that following oxidative injury, that 24 hours of oxidative injury, that's sufficient to activate this death caspase 1 in our control flies. But in our VHA44 knockdown flies, we see a reduction in DCP1 staining. So we then wanted to try to get at a cell signaling level what our VTPase knockdown was doing in these flies. So the c-Jun N-terminal kinase or JUNK pathway, it's a highly conserved pathway that responds to cell stress. And a previous lab using Drosophila in the imaginal wing disc, which is a developmental growth model of cell growth, had found that overexpressing some of these VHPA subunits lowered endosomal pH and actually led to JUNK activation. So we thought maybe our suppression was lowering JUNK activation in our flies. So we used a common reporter of JUNK activity in Drosophila called PuckLAX-Z, which is based off of this puckered gene, which is activated upon JUNK activation in a negative feedback loop. So when we look at this PuckLAX-Z JUNK activity reporter in our control uninjured flies, we see relatively low levels of JUNK activation in the intestine. But upon feeding with our oxidative stressor, we see an increase in JUNK activation. And when we knock down our VHA44 subunit, we see a decrease in that JUNK activation, as quantified here in our vehicle and our oxidative stressing. In parallel experiments, we also were interested in looking at innate immune signaling. And so we first just looked at the IMD pathway, the ortholog of NF-kappa B relish, and we found that just relish knockdown in enterocytes was sufficient to protect against oxidative stress in our animals at a similar level as vacuolar ATPase knockdown. So this led us to want to test the requirement for vacuolar ATPase in activating both local and systemic innate immune response in our flies. So we used a drosomycin GFP reporter, and we see with oxidative stress, we see strong drosomycin GFP activity as expected. And when we knock down VHA44 in the enterocytes, we see a reduction of that local effect. And then at a systemic level, we use qPCR to look at a couple of antimicrobial peptide genes in Drosophila, ataxin C and DPTA, and found that knockdown of VHA44, specifically in the enterocytes, was sufficient to suppress these AMP expression. So we then turned to look at TAC1, which is a protein that can signal both through the JUNK pathway as well as the IMD pathway, as maybe something that could control both of these pathways. When we overexpress TAC1 in our oxidative stress condition, we actually see that it sensitizes flies to our oxidative stress, and that when we knock down VHA44 at the same time, it's actually able to rescue back to wild type levels of survival. So we next want to test if TAC1 itself was able to drive JUNK expression in our model. This time we used phospho-JUNK stain to look for active JUNK signaling. And again, we see in our oxidative stress condition, when we knock down VHA44, we reduce JUNK activity. However, overexpression of TAC1 was not sufficient to enhance JUNK activity by itself in our system. So that led us to want to look at IMD signaling in the context of TAC1 overexpression last week. So we looked at our ataxin C and DPTA in this oxidative stress condition, and we found that overexpression of TAC1 drove systemic expression of these antimicrobial peptide genes, and that knockdown of VHA44 was sufficient to suppress this effect. So overall, that leads us to a model where we see Our oxidative stressor drives lysosomal acidification, which appears to be important in driving both a strong junk signaling as well as IMD signaling in response, possibly through TAC1, especially in the case of IMD signaling, which leads to interior intestinal barrier dysfunction, cell death, systemic immune response, and eventual organismal death. So going forward, we want to look at if this increase in enterocyte lysosomal acidification is sufficient to drive junk and IMD signaling by itself, and if TAC1 is required for that. And then we also wanted to move into mouse and organoid models to see if that translates into these models. And judging from all the pretty pictures from the Ingevic labs, maybe I should reach out and talk to them about organoids. But with that, I'd like to uh, thank my lab and my funding sources. And hopefully I didn't forget to say too much without my notes. Thanks.
Really, really great job. Very interesting talk. We have time for maybe one very quick question. I don't know, Mindy, if you would like to ask one of yours.
I have a question for Dov.
Yeah.
So, okay, how specific regarding the MAP kinase pathways do you think that is to DNK? Have you checked others such as ERK or P38 or?
Yeah, so we did briefly look at ERK and didn't see a change, but in P30 we haven't looked at P38 yet, which could be a good idea. It's a little bit less involved in Drosophila intestine than junk generally, but yeah, it's a good idea.
Yeah, okay, because usually all those kinases have like such a mechanism that maybe ones are compensating the others. So just a suggestion.
Yeah, thanks.
Well, Doug, again, thank you. And there are
Thank
some
you.
more questions in the chat, but I'll turn it back to Christina to introduce our next speaker.
Okay, so our last speaker is Anna Tingler. She is doing her research in the Medical University of South Carolina. So it's a pleasure to have her here talking a bit about how microbiota changes is disrupting the mucus barrier in patients suffering from cystic fibrosis.
All right, can everybody hear me?
Yep, we can hear you.
Perfect. Thank you so much for that introduction, and I will get right into it. Cystic fibrosis is characterized by a mutation in chromosome 7, and chromosome 7 codes for a protein called the cystic fibrosis transmembrane conductance regulator, or CFTR, and that regulates chloride transport in the body. Mutations in CFTR cause dysfunction or inactivate CFTR, leading to improper chloride and bicarbonate anion secretion. This leads to the accumulation of thick, sticky mucus lining the airways and other organ systems. Cystic fibrosis affects many people. In fact, 1 in 25 people carry a defective gene for cystic fibrosis, with 40,000 people living in the United States with cystic fibrosis, and 105,000 people worldwide have been diagnosed with cystic fibrosis. This results in 1,000 new cases each year, and unfortunately, this is a chronic condition with no cure, which really underscores the importance of continued research into cystic fibrosis. Primarily, cystic fibrosis is known for its disruptions to the lungs. Here you can see immunostaining of a biopsy of the airway in a control, non-CF individual, and a cystic fibrosis individual. And you can see from the brown immunohistochemical stain of mucus that there is that classical manifestation of mucus overproduction in CF individuals. Cystic fibrosis is also known to cause gastrointestinal distress, leading our lab to focus our research on cystic fibrosis in the context of the gut. Shown here is an Alcian blue staining of small intestinal biopsies between control, non-CF individuals, and CF individuals. You can appreciate the accumulation of mucus stained in blue in the CF small intestine, consistent with the phenotype we see in the lungs. However, CF is widely understudied in the large intestine or the colon, so my lab has decided to tease out this knowledge gap. With the recent development of CFTR modulator drugs, the life expectancy of these patients with CF has increased substantially. However, CF individuals are still prone to larger, more aggressive colon polyps. They have a 6-fold increase in developing colon cancer, and 50% of cystic fibrosis individuals over the age of 40 will develop adenomas. So to begin to understand cystic fibrosis in the context of the colon, we performed immunostaining of human colonic biopsies graciously given to us by our collaborator, Dr. Mary Beth Nicholson. Here in the staining, the mucus can be seen in blue. When comparing control non-CF tissue to CF tissue, we surprisingly see similar levels of goblet cells per crypt. To characterize the mucin profile of CF individuals, we performed immunostaining for MUC2, which is responsible for the secretion of the gel-forming mucus layer. Our staining found that MUC2 is the most dominant mucin found in the colon, followed by MUC1. MUC1 can be seen in green, and MUC2 can be seen in purple. Here you can appreciate the MUC2 levels between non-CF individuals and CF individuals are rather similar, which once again was surprising to us. Quantification of these images also showed no significant difference between MUC2 levels in non-CF individuals and CF individuals. To help us validate our results above, we turned to RNA sequencing data due to its larger sample size. We found from our analysis of this dataset that validates our staining shown previously. The bulk RNA sequencing shows that no significant difference of MUC2 expression between non-CF individuals and CF individuals was noted. However, something interesting caught our eye during our next set of staining. Highlighted in yellow is staining from CF individuals that shows a significant decrease in mucus levels. But what is different about these cases versus our other population of CF individuals? We found that these individuals have been taking antibiotics. CF individuals taking metronidazole and rifaximin have a decrease in mucus, and amoxicillin, on the other hand, shows a depletion of mucus in non-CF individuals. You can also appreciate this depletion of mucus in CF individuals taking antibiotics by immunostaining, specifically highlighting the decrease in MUC2 stained in purple. Quantification of the staining revealed a significant decrease in MUC2 relative fluorescence in CF individuals taking antibiotics versus non-CF individuals. To reciprocate these findings in a mouse model, we gave wild-type mice 4 millimolar antibiotics such as metronidazole and amoxicillin in drinking water. We then performed an Alcium blue stain of the colon and found that antibiotics are associated with depleted mucus as indicated by the shrinking colonic mucus layer in the antibiotic-treated mice. Quantification of the staining revealed a significant decrease in mucus layer thickness of the antibiotic-treated mice versus our water-controlled mice. To begin to uncover how antibiotics directly affect cells, TD4 cells were plated down, DAPT was added to drive goblet cell differentiation, and these cells were treated with antibiotics shown to deplete our mucus in our clinical samples. And then MUC2 gene expression was determined. We found that these antibiotics do not suppress MUC2 expression at 24-hour treatments and 48-hour treatments. We also wanted to validate this effect in human-derived colonic organoids. The results from this experiment indeed upholds our previous experiment showing no suppression of MUC2 from antibiotics alone. We also wanted to evaluate if goblet cell function and mucus expulsion is being affected by antibiotics. To do this, we added a stimulant to T84 cells that causes the release of mucus by goblet cells. That release of mucus can then be measured and quantified. We found that T84 cells stained with Jacqueline, a lectin that recognizes all mucus sugars, and treated with a mucus stimulant, significantly expel less mucus in antibiotic-treated conditions, including both concentrations of amoxicillin and metronidazole. As we continue to uncover the role antibiotics play directly on goblet cells and mucus expulsion, we wanted to explore how the gut microbiota could be involved in the decrease of mucus seen in CF patients taking antibiotics. For example, the gut microbiota is able to secrete bacterial-derived compounds that in turn could be involved in influencing mucus secretion from colonic goblet cells. When antibiotics are delivered to the gut microbiota, it alters the gut microbiota present and therefore alters the bacterial-derived metabolites that are secreted. These altered metabolites could influence the significant decrease in mucus production seen in CF individuals. To uncover how the microbiome is altered or changed in non-CF individuals versus CF individuals and CF individuals taking antibiotics, 16S sequencing was performed on human stool. We found that in non-CF individuals we had dominant populations of Firmicutes and actinobacteria. But when we looked at the stool of CF individuals, we see a striking change with blooms of proteobacteria and Bacteroidetes. And we even noticed slight differences in the microbiota of CF individuals taking antibiotics. To investigate more closely, we wanted to see if our CF individuals taking antibiotics were lacking types of microbes responsible for increasing mucus production. To start, we noticed a significant decrease in bifidobacterium in CF individuals taking antibiotics when compared to the other groups. Recent advancements in research have highlighted bifidobacterium dentium to be involved in increasing mucus production. We have also noticed a significant decrease in Blauta in CF individuals taking antibiotics when compared to the other groups. Recent advancements in research have highlighted Blota coccoides to be involved in increasing mucus production. We also noticed significant decreases in other pro-mucus stimulating bacteria, such as those bacteria belonging to Calinocella and Aristopes. To dig deeper into how antibiotics could impact bacterial-derived metabolites, specifically in the context of pro-mucus stimulating bacterial-derived metabolites, we curated stool bioreactors. Stool bioreactors involve taking patient stool and growing it up in an enriched media. This allows for the growth of the microbial communities that can simulate a patient's gut microbiota. We then delivered antibiotics to those bioreactors. After growth or antibiotic treatment, we spun down the bioreactors and isolated bacterial-derived metabolites secreted into the growth media. The bacterial-derived metabolites were then added to cells, and MUC2 expression was quantified. Bioreactor metabolites from control individuals that did not receive antibiotic treatment were able to elevate MUC2 expression. However, antibiotic-disrupted bioreactor metabolites from control individuals failed to elevate MUC2. Staining of MUC2 in the pink revealed that the bacterial-derived metabolites from control individuals not treated with antibiotics were able to bolster MUC2 levels from our DMEM control. However, antibiotic groups failed to bolster MUC2 levels. Bioreactor metabolites from CF individuals that did not receive antibiotic treatment once again were able to elevate MUC2 expression. However, antibiotic-disrupted metabolites from CF individuals failed to elevate MUC2. Staining of MUC2 in the pink revealed that the bacterial-derived metabolites from CF individuals not treated with antibiotics were able to bolster MUC2 levels from our DMEM control. However, antibiotic-treated groups, once again, failed to bolster MUC2 levels. Therefore, we can uncover that the gut microbiota can secrete compounds that elevate MUC2. But when antibiotics are administered, they are able to reduce bacterial-derived pro-mucus stimulating metabolites. To investigate this further, we collaborated with our collaborator, Dr. Thomas Horvath, and we wanted to perform non-targeted mass spec to determine what metabolites were present in control stool versus CF stool and CF stool taking antibiotics. We found that amino acids involved in mucin synthesis are decreased in CF individuals taking antibiotics. For example, we observed significant decreases in serine among CF individuals taking antibiotics, but we also observed significant decreases in leucine, threonine, glutamine, glutamate, and neurotransmitter GABA. So to uncover the ability of these specific metabolites to bolster MUC2 expression, a cocktail of amino acids, fatty acids, and bile acid TUDCA were added to the cells. TUDCA was included as previous studies have shown it to have the ability to increase mucus. Quantification of MUC2 revealed the ability of these cocktails to bolster MUC2 expression, potentially highlighting their importance and significant lack of presence in CF individuals taking antibiotics. Taken together, our working hypothesis suggests that antibiotics could be playing a direct role on goblet cells and having an effect on bacterial-derived metabolites from our gut microbiota. To this point, our data suggests that antibiotics are able to affect the ability of goblet cells to expel mucus, as well as deplete the normal gut microbiota, thus decreasing specific bacterial-derived metabolites found in the gut. Working in tandem with each other, the resulting effect is a decrease in the colonic mucus layer and mucus secretion. A decrease in the colonic mucus layer has shown to potentially cause inflammation and cancer, and therefore my hopes are that I will be able to add back these amino acids and other short-chain fatty acids to potentially bolster the mucus layer back to pre-antibiotic levels, which could potentially inhibit inflammation and inhibit cancer production in these patients with cystic fibrosis. And so with that, I would just really like to thank my lab for all the support, thank all my sister labs, thank all of my wonderful collaborators. This would not be possible without them. And I just want to give a special shout out to PISA for giving me the amazing opportunity to speak here today. And with that, I will take any questions.
Thank you very much, Anna, for your amazing talk and for all the work that you have shown that you have done and there's behind that project. So do we have any questions in the audience? We have some time for them. There are some questions in the chat already, I think.
Anna, that was phenomenal. As always, congratulations. That was super, super interesting. I might be asking a silly little question, so please enlighten me. But of course, you showed these phenomenal pictures again. from the Engavik of the colon disruption and decreased mucus production after the antibiotic use. Did you happen to also check out the lungs of either the patients or the mice to see if mucus production was altered? And would you fear or like have a hypothetical of for the future directions, which were super cool, by adding those supplements in order to kind of restore the mucus production? Could that have any side effects for the lung?
So, that's a great question. We have not tried our, like from the mice, we didn't collect the lungs. Um, we want to try to specifically localize those metabolites to the colon, and we would do that by rectal administration. So with that, I would hope that we would minimize the side effects that we would see of like potentially increasing the mucus that we don't want to increase in the lungs. So, I hope that answers your question.
Yeah, that's super fascinating. Thank you so much.
I'm trying to find the other ones in the chat, but if someone could read them out to me, that would be great.
Yeah, we still have some time for a quick question,
Let's see.
or I
I
can
think,
ask you, do you think that all the effects that you have observed with the goblet cells function, if you introduce like periods with antibiotic-free treatment, let's say, do
yeah.
you think that these effects could be restored?
Yeah, so I think that's definitely possible, because after antibiotic treatment, your gut microbiota will, reform and you'll have like a healthy microbiota. So I think it's possible that if you introduce periods of time when you don't have antibiotics, that those pro-mucus stimulating metabolites could come back that we have been missing and potentially restore the mucus layer. However, in these patients with cystic fibrosis, they usually are on chronic antibiotics. And so I think that's really contributing to this pronounced effect of their decrease in the mucus layer. And so they don't really have the chance to, take days off of antibiotics.
Yeah, yeah, you're right. That's the approach, the therapeutic approach is that. But yeah, maybe then you should consider maybe like any probiotic supplementation or something like that without like that could kind of
Yeah,
restore the adverse effects of the antibiotics, but not affecting the aim of the therapy.
right, for sure, for sure.
Okay.
Let's see. So Cole, you're asking about if we see any overlapping genes like that are unique to cystic fibrosis patients. Am I correct?
Hi, Anna. Sorry, I'm driving. Can you hear me?
Yeah.
Okay. So yes, so ampicillin, is a derivative of the psyllin family. And so I
Yep.
was wondering if like maybe you see similar phenotypes in patients that don't have cystic fibrosis but are taking long-term like penicillin or any other of the sort of the derivatives. And if you do or don't, do you think that there is an upregulation or downregulation of genes that are specifically unique to CF patients that you don't see in the other group?
Yeah, so that's a great question. So we only got samples from like the patients that I showed you, like the antibiotics that they were on. So I'm unaware if, there's any changes in patients that may have been on penicillin or derivatives of those antibiotic types. Um, I it would be really interesting to observe if there's any interesting, like unique genes to CF patients. I am unaware of any that like, pops out to me, but yeah, that would be really interesting to look at.
You did a great job. Thank you.
Thank you.
Okay, that was a really great discussion. So I think that with that we can finish this session. Thank you very much to everyone for attending and for all your participation and for the amazing talks that we have had in this session.
Yay, first session complete of PISA Day 3. We had a 5-minute break worked into the program. As of now, we have 2 minutes left of that 5-minute break. So if you want to go run and go to the bathroom, grab a drink or something, we will start right at 1:40 with a career special talk given by Dr. Chloe Kirk. And Chloe, if you wanted to test your screen or sound or anything, feel free to do so.
Oh, yeah, sounds good. Maybe I'll try it and go ahead and share my screen just so I've got that sorted. Let's give this a go. All right. Press play and just tell me if you're able to see it. Once I press.
I can see it.
How about now?
Perfect.
Fantastic. All right, I'm gonna press stop sharing. Maybe.
You're welcome to keep it up too. That's totally
Okay, cool.
Thanks for coming here today.
Thanks for having me. Excited to be here.
Now we have an awkward 1 minute of Zoom break. Love that. Okay, well, that was a really quick 2 minutes. But welcome everyone to our last special talk of PISA on our last day, PISA day 3. So my name is Sarah Dooley. For those who don't know, I have a PhD from the Medical University of South Carolina, and I'm very excited to welcome you to Finding Your Fit in Science careers. So a lot of trainees who are excellent at science are kind of wondering what happens if I don't stay in academia, what else is out there. So today's speaker, Dr. Chloe Kirk, has actually walked that path and is amazing science communicator and person who really accelerates what it's like to navigate looking at different career paths within science that are not academia. So Dr. Kirk earned her PhD in biochemistry and molecular biology from the University of Miami in 2024. And it seems like during her PhD, she loved being a science communicator and was very actively involved in science outreach initiatives. And you'll see that she is an amazing science communicator if you haven't seen her yet. She has a large following on social media. She's worked in patent law. And most recently now she's joined Biotech TV, where she's breaking down some very interesting science into educational segments. And yeah, so I'm a huge fan of hers, and I'm certain you'll leave this talk with some better understanding of skills that you already have that are transferable and maybe a clear idea of what's possible for your science career. So please join me in welcoming Dr. Chloe Kirk.
Fantastic. Well, thanks so much for having me. Really excited to be here, Sarah, the whole team. Thanks for organizing. I'm really excited to be giving this chat today because I think this is a really undervalued topic. I mean, I'll be speaking about my own personal experience here as well, but there isn't much that we talk about in academia about life outside of academia. So I think this will hopefully help at least a couple of you. So yes, what I'm going to be addressing today, what's beyond academia? The looming question no one talks about in school. A lot of this will be from the first person of my experiences leaving academia. I left the lab in 2023. And then navigating science career options. And most importantly, because this is the question I probably get the most frequently, is how to find jobs outside of academia. The dreaded networking, yes, we will be going through it, and hopefully networking will sound a little less scary to you afterwards. And then the final piece of information will be what to do once you're done with this conference. All right, let's start with who I am. Hi, I'm Chloe. I got my PhD in biochemistry back in 2023 at the University of Miami. Before that, I did my bachelor's at the University of Wisconsin-Madison up in the Midwest. I was a dual major in molecular biology and like pre-law. And I always love to include that before all of this, I was in high school at a small charter school in Minnesota. My graduating class was 27 people. And we really did not emphasize science. I took all the science classes that were offered by like junior year. Did not think science was for me at all. And it wasn't until I first stepped into a lab that I realized how cool science actually was. So a bit of what I've been doing most recently after I left the lab, I moved to DC, where I joined a big law firm here in patent law. And I did that for a couple of years, and now I've switched over to doing science communication full time. As was alluded to previously, I was doing science communication a little bit throughout my PhD, but it was always like a passion project, and I feel very fortunate to be able to do that full time now. All right, very important elephant in the room. Why am I talking to you today? Why me and why not some other professor that's on there? So I've been in your shoes quite recently, matter of fact. I have explored a lot of science careers. I started looking, gosh, maybe a second or third year of my PhD when I realized I didn't want to go through the route of postdoc, professor, all of that. And the traditional industry career path of, applying to, maybe doing a postdoc first, then applying for industry positions also didn't float my boat. So I chatted to, God, I think it was 98 different people over the couple of years in all sorts of careers. And I'm very vocal about chatting about this online, as also was alluded to. So the first concept that I really want to hit on the head is, what does it actually mean to be a scientist? This was a conversation I've had so many times with friends, both in and out of the field of academia. And if you just Google this, what most of the Merriam-Webster, Oxford Dictionary are going to pop up, an expert in one or more of the natural or physical sciences. Now, you'll note that definition does not include academic, professor, or researcher. And this is something that I had an identity crisis when I left grad school, because I left academia thinking I shouldn't be calling myself a scientist anymore, since I'm not actively working at the lab every day in academia. And it's a conversation that's taken me so long. In fact, my Instagram bio was Chloe the Scientist. I came about this close to changing it to something completely different because I felt like kind of an imposter calling myself that still. And it's this term that I think academia has taken and made it into this thing where if you leave academia, you're no longer allowed to call yourself this scientist. I think we need to get rid of that notion altogether. There's an academic researcher, very important, what a lot of you guys are doing right now, that ends up becoming the academic tenured professor. But we need scientists in all sorts of careers, not just academia. And so that's the first big thing I want to dispel from you guys, is that scientist does not mean that you need to even work necessarily in a lab. There are so many cool scientists doing so many cool things that aren't picking up a pipette anymore. So number one, transferable skills that you have from science. When you think of, okay, what am I going to be taking out of my undergrad lab experience or my PhD lab experience? You're probably going to be thinking, okay, I learned how to do Western blot, or I learned how to do microscopy. But there's so much more you need to do than that. And the first thing I challenge you with is to sit down and write a list of every transferable skill you have. I've written some of the generic ones that I started with when I was thinking of, okay, what did I actually learn from my PhD that wasn't how to pipette something? I'm critically thinking and analyzing data, efficiently reading complex topics. If you remember for the first time you picked up a science paper, your freshman year of college, it probably took you a few hours to get through. And flash forward to, say, if you're at the end of your PhD, you might be able to read through a paper now in 20, 30 minutes and get through the key ideas and understand what each of those figures mean. Really important skill that we often just kind of blow by because we become used to it as second nature in academia. Multitasking. Think about the amount of experiments that you've done at once, or the experiments that you're also trying to write a grant, and at the same time you're writing a paper, preparing for a conference, excuse me. You've got so many different things going on at once. You need to give yourself some credit that you're actually doing a lot very well. Also, time management, organizational skills, problem solving, etc. One I really want to touch on is this perseverance or grit. You're going to notice this image, this YouTube video next to me. It's called Grit, the Power of Passion and Perseverance. If you've not checked out this YouTube video, I've actually done the QR code. Please scan. Don't watch it right now, but after this talk, go and watch this video. Or maybe after the conference is done for the rest of the day. It is so powerful. It's not specifically about science, but it's about a concept that we do every day in the lab. And it's the idea of grit, the power of passion and perseverance put together. I hope that you all come out of this as with a new idea of what you've gained from your research, because there's so much more you've gathered than just how to I don't know, make a spreadsheet look nice. Teamwork, adaptability, analytical skills, go on and on. All right. Now, the thing that I think is the most important, and what I hope to spend the most time on chatting with you guys today, is really just breaking down the nuts and bolts of how to find your science career fit. I've broken it down into 4 steps, and we'll go into each one. The first couple will sound really easy, then the third one's going to get a little scary because that's where that dreaded term networking comes in. And then you'll notice that I put applying for the job at the very bottom of this list, because if you're going into the job hunt with just starting applying for jobs, fortunately in today's market, that's not going to get you anywhere. So it's very important to leverage this big scary term network, which I know a lot of us went into science thinking that we wouldn't have to do, but yes, you're going to have to. All right. Aligning your interest with the 9 to 5. What about your work do you enjoy? And then what hobbies don't currently fit into your work that you do enjoy? So some examples, I mean, if you like doing bench work, maybe hobbies that don't fit in, say if you like writing a blog or I can't speak today, riding a bike. I put some lists on the side of examples because I don't want you to say it's just specifically science skills on this list. When I was doing this list, and this was my second year of my PhD, I wrote down everything under the sun. I wrote down that I like to bake. I like to travel. Yes, I like to communicate science. I'm sorry, I've got a train passing by right now. But it's important to write every part of these hobbies and anything that possibly interests you onto this list. Because at the end of the day, you're not going to find a career that gets all of your interests together, but having them all on one piece of paper will really help you with step 2, which is finding a job matching those interests. And this is when the age of technology, Google, ChatGPT is really helpful. You can probably do this right after this conference tonight. Don't think about it too literally. Just start typing into Google, ChatGPT, your interests and finding jobs that align with these interests. And a very important key here is don't limit yourself to science-sounding jobs. I know a lot of us, we want to go for the medical writer, the things that we think sound very sciencey, but I cannot emphasize enough how valuable it is taking your science skills and applying them to jobs that aren't traditionally for scientists. Because you have, and I'll go back to that slide a couple of slides back, you have so many skills that traditional people that if they didn't go through a science education don't have. And being able to interact with people that have those different sorts of skill sets is invaluable for all sorts of things. I mean, one that really comes to mind right now is if you think about policy. All right. So let's say you've done all of that, you've started plugging into ChatGPT, and you've got this long list of jobs that you don't even know what to make heads or tails with. This is when the scary part comes in. And we're going to make this less scary, hopefully by the end of this all, is holding down to the perfect fit. So how I started this, and I was doing this all from 2nd to 4th year of my PhD, is I took that list from step 2, and it was probably 20, 30 careers, and I plugged it all into LinkedIn. I would literally look up the job title and then start scrolling through people I had mutual connections with. Another option to use, and I've highlighted this down here, is the school alumni database. Bigger schools especially, you guys have really robust alumni databases, and it doesn't just need to be your PhD or grad school program. I was using my undergrad, UW-Madison's a ton as well. Also reaching out to mentors and advisors that would be able to connect you with certain interests is really helpful. And always bonus, if you have some sort of commonality to facilitate that introduction, if it's a mutual connection like on LinkedIn, if you went to the same school is really big, some sort of club or say PISA or some sort of association that you guys have as a related group. The big thing, networking is just learning from other people's experiences. Go into it thinking it's just a conversation and not this big, scary, I don't know what I'm doing. I've got to figure out how to network in the next 24 hours. So networking made easy. I'm going to go through the securing the interview, going for the interview, and after the interview. There's 3 different things, and they all require a little bit of practice to get right, but every time you do it, you'll get better and better. So securing that interview, short and sweet. Like I said, you want to mention any commonalities if you have. I even included this is literally, I found this in my notes. This is something that I had ready to go whenever I was reaching out to someone on LinkedIn. I would add, say I put PhD student at the University of Miami interested in pursuing a career in science writing. Add a little extra sentence there saying like, we're both part of PISA, I noticed, or something like that. You also want to minimize the back and forth. So you want to give them everything they have up front in the short little caption. If they get back to you and say, yes, love to meet, give them everything right away. If they're asking for your availability, say, I'm following up in an email, send it all together. You want maybe 2 or 3 emails back and forth before you've got a time and date secured. All right, going for the interview. So something, this kind of goes into securing the interview, but it's interesting now being on the other side and getting people asking me to chat with them about various career paths on how often this isn't done. So it's going to sound basic, but bear with me. calendar invite with a meeting link. Especially nowadays, we're doing everything virtually. Have it all really easy for people to get to. Calendar invite's also really nice because you can have it auto give them a reminder that they've got the meeting with you, say 30 minutes before the event starts. So you aren't having to worry, oh gosh, are they going to remember this? It's already on their calendar. You're good to go. Making 2 sets of questions. Now, there's going to be, obviously you're chatting to them about the job they're doing. In this case, the example I was giving is science writing. And hopefully you'll have some sort of list prepared of questions, how, if you think science writing's the right fit, or what day-to-day life might be in science writing, kind of like generic questions of, okay, would I like doing this job? But the other part that's really important to focus on is the person, because at the end of the day, you're chatting with another human being. It is a conversation and a two-way street. So you want to make sure you've done a little bit of background homework on them. And we're not talking extensive here. We're talking like, look them up on LinkedIn, see where they went to school, what their major was. Did they do any clubs that you thought were interesting? Did they do any travel you thought was cool? Do they have any interesting publications out there? Just do a little bit, like 5-10 minute Google search on them. Now to remember, it's just a conversation. Often I've noticed now when I'm on the receiving end of getting these requests to chat, they will dive right into, hi, my name is Chloe. I want to know this, this, this. But I want to know a bit about you as well. So make sure you give a little bit of an introduction of who you are, why you're chatting with them, even when you get on that call to begin with. Last, time is money. And I say this both in terms of you want to show up early to make sure that you're prepared, ready to go for the meeting, but also be mindful of their schedule. If you have a ton of questions, you notice you've got 5 minutes left of the 30-minute block you've scheduled, and you're like, oh gosh, there's no chance I'm going to get through all of these. Just pause for a second and say, hey, I'm having a really great conversation with you, and I've got quite a few more questions than I expected. Do you have another 10-15 minutes in your calendar? Most of the time, they will. Most of the time, they'll have a ton of time. If they don't, they'll say, and often you'll be able to follow up via email or even schedule another call. But showing that you're being conscientious about their time, another very underrated skill to have. Finally, the follow-up. Oh, I can't see this on my screen. Let me see. I might just have to, I've got all the people's photos on the side, but that might just be how it is. All right, we're going to do follow-up within 24 hours afterwards. I'm shocked with how many people don't do this, and it sounds really easy or scary, depending on how you want to put it. But literally just send a one-sentence email. Thank you so much for your time. I really enjoyed learning about XYZ. Also in this email, very easy to say, ask for people to connect with. Hey, I really loved learning about the medical writing program that insert university or insert company. Do you happen to have any connections that work at that place you might be willing to introduce me with? That very quickly turns into this like tree branching effect where, yeah, you might start with reaching out to 10 people cold on LinkedIn and 5 of them respond. But of those 5 people that you chat with, if each person's able to give you 3 more connections, that's 15, so on and so forth. Another thing that I didn't start doing until I started doing, I don't know, 10, 15 of these interviews or networking calls and started losing track of everything is write down everything you learned within the first 24-hour period. I'll often, like I have it in front of me, like a notebook or a notepad to write down all my notes I'm learning. But it's very easy to just like leave that to the side and then do the next interview and then you just have this collection of notes from all these talks you've had. Take 5 minutes after that call or maybe at the end of the day from that call and write down not only what you learned, but also the takeaways. Like, okay, could I actually see myself in a career in science writing? And then I say do this online because then keep a list of everyone you meet, and then you can have a link to each of those notes for each person you meet. So I have a gigantic Excel spreadsheet with the names of the people, the companies, and then I just have a link to when I met with the person that I'm able to look back to. And this is really important for step 4. Which is then you apply for the job. So ideally, you're going to be doing this in, anywhere from the first year of your PhD to the 3rd. Earlier is the better, honestly, because the more time you give yourself to do this, the less stressful it seems. I waited till my 2nd or 3rd year, so it's completely fine if you're doing this on the later end, or even 4th or 5th. Once you've started compiling this robust list of contacts, then you get to go back to it once you've done all the heavy lifting, let's say. So it depends on the sort of company role job you're looking at. You could be applying to things anywhere from a year out from your PhD or finishing undergrad to say 3 months. That will be something you'll have to figure out as you're chatting with the various various connections you're making. Once you've gotten to that point, I don't know, put a calendar alert on your phone to say, okay, we're at that year mark, it's time to actually apply for these things. You're going to reach back to all those people you've networked with and see if they have, I mean, to be frank, you're asking nicely, is there a job opening that you know about? Now I've heard and I've gotten a lot of people that will just reach out, cold email and be like, hey, really interested in chatting with you and getting a job. And they'll kind of cram it all together in one. You don't necessarily want to do this if you can help it, because it then makes the networking call look transactional rather than more of an informational build. And that's a really big difference that you should be striving to make it seem more of like an informational interview, that later on then you get the benefits of what you call the networking part of it all. Also important to join groups and communities that have been recommended to you by those people you've been chatting with over the years. Because to be frank, LinkedIn and a lot of the other job sites nowadays, they are hit and miss when it comes to looking for jobs. And so a lot of them actually get posted through various community Slack channels or group chats for very cool opportunities. I was in a science policy community for a couple of years in grad school, and that was exclusively where the science policy jobs were posted. Like if I looked up science policy job on LinkedIn, it wouldn't happen. Also having a master resume and CV that you can modify for different positions. I'm sure you've heard this sort of thing for CVs, especially in academia, that you want to have a running list of everything you've done, but also having a master resume so that you're able to easily apply for a job when you see it's available. And it's even more important when, say, you've reached out to someone you've networked with, they're like, oh yeah, I actually do have this job that we put feelers out for, send me your resume. You don't want to be responding to them weeks later. You want to be responding to them within the same day. Finally, it's the thing that no one likes to hear, but it is the reality of the situation. Who you know beats your qualifications. You can have, and I've actually seen this very interesting statistics, because men are much likely to apply for jobs that they have less qualifications for than women. There's usually I think the study is that men will apply for something if they've got 5 out of the 10 qualifications. Well, women will usually wait and see only if they've got 8 or 9 will they apply for the qualifications. And there's so much to be said about your excitement and willingness to learn over actually coming to the table with those qualifications you have. In the case when I transitioned to patent law, which is a notoriously difficult transition because you're going from academic bench lab setting to a law firm where time is money, in the sense of you need to be billing your hours, you need to be reading very efficiently and being able to translate that science into law. And I didn't have very many qualifications going into that. One of the things that made me stand out and why they said they hired me was because I showed a willingness to learn. So I think that's like the number one thing you should be telling anyone, especially when you're applying for jobs. Oops. All right. And then going back to a little bit of my own life story, though, is there is no one right fit. As I alluded to, I went from academia, then I went to patent law, and now I'm in science communication. Careers are never linear, and that was something you will hear over and over again as you're chatting with people. And your interests will change. All experience is valuable. It's just how you decide to word it and how you decide to present that narrative. And to be frank, don't use ChatGPT for a lot of things, but ChatGPT is very valuable for figuring out how to word experiences in the right way to fit a new job application. So say when I was applying again for that patent law job, I didn't have any patent law background. I had my lab experience and I presented a couple conferences. So what I did was I harnessed my science communication skills of learning how to present, speak, different audiences, how to translate my science to other people. And I reworded that into, okay, well, I know how to translate science to the public. I bet I can learn how to translate science into legalese. All right. Then some other big just career tips that I wish I'd heard sooner, honestly. Practice science communication. All right, I'm going to say it one more time. Practice science communication. And I'm not just saying go present at a conference. Like, that is awesome and that is great experience and everyone should be doing that. But you also should be challenging yourself to talk about your science, to talk about science in general with people outside of the field of science. Now, and I should have put this actually in an earlier slide, there's so many different ways to do this. I'm just going to list a few here, is that you could do volunteering at a local science school, say like do an after-school science club, volunteer at a science museum. Most cities have science fairs, state science fairs. Like when I was in South Florida, they were always looking for volunteers, science experts to come judge science fairs, because not only was it you're kind of using your science expertise, but to be frank, the kids get a real big kick about actually meeting a real scientist who's judging their research. Yes, you can practice it online. You can post on social media like I do, Instagram, TikTok. If that's not your cup of tea, you can also write blogs. Tons of different societies provide opportunities for student scientists to practice writing communication by writing blogs about different science topics of their various associations. Any way you want to do it, it's possible. There's ways to do it virtually too, like Skype a scientist. I think I've mentioned a few others on a couple slides later. Think outside of the box. Your ideal job fit may not actually have a job description right now. And that was something I figured out when I left the lab. I really liked doing science communication, but I couldn't find a specific job that was really ticking all those boxes. But you can make it a reality. It's not going to happen overnight, but going back to that network that you have, as you start to signal to other people what your passions and your interests are, eventually doors will start opening down the line. And this was really cool. So yesterday, actually, 24 hours ago, I was in Chicago randomly, and I got to chat with the Nobel laureate for the Medicine Physiology Prize, Dr. Fred Ramsdell. And when I asked him, what was your advice for students interested in following your footsteps? He said, combine your passion with something useful for humanity, and you can't go wrong. And I loved that message, because fundamentally, at the end of the day, you're going to find a lot of things you're passionate about. And if you're able to find it with passion, mixing it with something that doesn't need to benefit all humanity, right? But say the basics of science research. At the end of the day, you are going to be helping a certain patient population out there treat some sort of disease. And as long as you've got those two things together, I don't know. He seemed very happy and he won a Nobel for it, right? Lastly, everyone has lab experience, but figure out what else you bring to the table. And I think this is something that a lot of my age, so recent PhD graduates, are dealing with the hard way, because we've been told for so long, okay, if we work in a lab, we figure out that specific microscopy technique, we're going to be set in careers for our lives. But there's a lot of scientists nowadays with a lot of lab experience, and we need to start figuring out how to make ourselves separate from the rest of the group. So what else are we bringing to the table? All right, now what to do after this conference? First and foremost, update your social media presence. If you don't have a LinkedIn, get a LinkedIn. And it doesn't need to be perfect, but put every single thing on there. And I'm even talking down to like your high school volunteering. Why do I say that? People look at LinkedIn way more than you'd imagine. As you get into the networking stage, which hopefully all of you do, you'll start going through people's LinkedIns as well. And you never know what sort of commonality you'll have unless you throw it all on there. So throw those certificates or the awards you won in high school or college. And also focus on beyond LinkedIn. If people Google your name, what are they going to see? You want to make sure even if you have an Instagram, a Facebook, etc. Because I have a private Instagram, I just make sure, one, it's private, and two, I'm not posting anything crazy on it, right? It's so easy to Google people nowadays before jobs, before you get the next stage of interviews. So just, be careful of what you post, which I'm sure you hear all the time, but it can also go to your advantage too. So if if you do some cool sort of presentation, say if you're presenting it like a 3-minute thesis, having that even on your LinkedIn or something, it will show up when people search your name. Make your interest list. That was step 1 of what I was talking about a few slides ago. And then challenge yourself to reach out to a certain number of people a week. My goal, because I'd started my 2nd or 3rd year, I challenged myself to meet 3 people a week, or reach out to 3 people a week, because not necessarily everyone will get back to you. But by doing that, the first 3, in the first few weeks I was doing it, took me a few hours to work up the courage. But once you start getting into that rhythm of, okay, it's Monday morning, I'm sitting down in the lab, I've got my Western block going, let's find 3 people that I think are cool. It will take you maybe 5 to 10 minutes, and it will expand your network dramatically. Finally, and this was the slide I was talking about where I had some opportunities for you guys, find a way to communicate your science. Speaking to classrooms, attending events to explain your science. So 3-minute thesis is a really cool option that a lot of universities offer. It is explaining your research in 3 minutes. Pint of Science, which is one where you can go to like a local bar and then they'll put on you talk about your science to a general audience. Volunteering local schools, science fairs, science museums. If you're feeling more ambitious as well, there's quite a few associations that offer, say, like small $500,000 to put on your own sort of science outreach event as well. And with that, I think I'm a bit ahead of schedule, so we've got more time for questions if we want. But yeah, I've left everything. You can connect with me on Instagram, TikTok, LinkedIn, email. Happy to answer more questions one-on-one privately too, if people would like to follow up via email. But otherwise, the floor is open.
Thank you so much, Chloe. I wish I would have had all that wealth of knowledge years ago, but I'm grateful I have it now.
I was to say, I wish I'd heard someone talk about this my first year of my PhD.
I know, that's why we need more people like you. Does anyone have any questions, or I can begin with one? Anyone have any dying questions?
I just wanted to make a comment. Chloe, you had a great talk for one, but also I like that you mentioned transferable skills because I don't necessarily know what I want to do once I graduate from university, if I want to go into academia or industry or whatnot. So I've actually been thinking about the list of transferable skills, but also seeing that my thought process or train of thoughts actually on the right track is pretty reassuring. So I thought you gave a great talk and I did learn quite a bit. So I appreciate you giving us your time today.
Thank you. That's great to hear. And I completely understand you about the transferable skills, because when I first started writing them on a list, I'm like, is this real? But it is.
I actually had a question kind of along the transferable skills.
Yeah.
So I am at the point where I am recognizing transferable skills, but I'd say that sometimes I have a hard time either writing that down on a resume, or sometimes I run into looking at job descriptions, and even if there's a transferable skill, I might not have clear, hardcore, actual experience in an industry setting or in a specific avenue, even though I know I'm like, I could do that. So, do you have any thoughts or advice on that?
Yeah, I think we are our own worst enemies, because I've come across that so many times myself. That I think because I've not done it in that specific setting, that it diminishes the value of the work I've done. And it's hard to say, or it's hard to hear maybe, that the easiest thing is really you just you've got to be your own best advocate, right? It's You're not lying if you say you've done it, even if it's not in the right setting. And I think especially for industry things, when you see something, if they say, a big one I've seen a lot is teamwork or managing different people. You do that all the time in a lab, even if you're not actually the lab manager, even the senior most PhD or postdoc. Think about every single interaction you have with other people around you is in some way teamwork and managing. So I think there's a difference, right, between like exaggerating something and you don't want to be making it up completely. But I think having a lot of faith in what you're saying, at the end of the day, you'll be able to explain it if they get to the stage of an interview. And if they read it and they don't think it aligns, then it doesn't go any further. So I don't have any concrete advice, much than just be your own advocate, and you know what you're capable of the best. So you've got to stand up for yourself.
Yeah, I think that goes a lot with the statistics on women applying for qualifications versus not, but it is helpful to hear as well again.
Yeah, I'd show my husband a list of the industry jobs I was looking at me a few years ago. They're like, oh gosh, like I only fit a handful of these. And he would actually, maybe this would be part of the advice, is find someone who's not in science, show them the jobs you're looking at, and see if they can match what you're doing in grad school, PhD, postdoc, to what those skills actually are on a piece of paper. Because I think a lot of scientists too, we diminish our own worth of what we do beyond pipetting.
Yeah. I think we have a question from the chat that I can read from Taylor. She said, really great talk and so relevant. Since a resume is so much shorter than a CV, how do you pick and choose what to highlight and what to include in the resume when applying to non-academia jobs? I would imagine it's very difficult than when applying to academia positions like we're trained for in grad school.
Brilliant question. I feel like that could be a whole other talk just in itself. And I don't have a great right answer for this, because I don't think there's any one right way to write a resume. That also goes back to the importance of using social media presence, especially things like LinkedIn, because you can kind of treat it like your CV where everything's on it. And nowadays, the very common thing is on your resume, you put like links, because most people aren't printing it. They'll be able to click on, say, your LinkedIn, and they'll be able to see everything else. But for picking what to highlight, I'd actually, and this is something that's worked very well for me, I'd actually use ChatGPT to help you with this. So what I did when I was looking at certain jobs is I took my CV and I then took the job description of either it was a link or I had to print out the PDF, and I'd throw it into the machine and say, okay, what of my skills and things I've got here are the most applicable to this job? They'd be able to narrow it down, and then from there it would be working on the rewording so that it matches the actual job description. But yeah, I'd use ChatGPT for that to be able to help you at least narrow it down. The other thing that is a big, I wouldn't say mistake, but I try to avoid if you can, if you're applying to non-lab jobs, is don't talk much about the lab work that you've done on the resume. I know it's awful to hear. That I had 3 different mentors have to tell me to take out more and more of my science research background, because I had like a whole section of the various different techniques I'd learned in grad school. But at the end of the day, especially if they're not lab people, they're not going to give one toss about it. And so thinking, I'd have a skills section actually, but I would focus more on those transferable skills we just talked about. Yeah, hopefully that was helpful.
The floor is yours, Maria.
Thank you, Sarah. And thank you, Chloe, for that like kind of eye-opening talk. Just because I'm in grad school right now. My PI actually just moved to UMiami, so go Canes, I think it is.
Yep.
Yeah.
I'm gonna stop sharing actually, because I can only see one person chatting at a time. Oh no. Is anyone able to get me to stop sharing? It's now stuck.
I don't know how to do that for you.
Oh no. Oh.
Yeah.
There you go. Perfect. So I
There
just
we go.
kind of wanted to ask, like, how did you know when you wanted to pivot? Because in like so many of my TAC committee meetings or just one-on-ones with other people, the options that are presented to me on a platter are academia or industry. And like no one really talks about the other alternatives, the other options, the pros and cons of each. You kind of just like learn through talking has been my experience. But when did you decide, I don't want to keep staying here. I just want to move to something completely different. And also learning those skill sets that helped you grow into where you are right now.
Yeah, fantastic question. There was no moment, there was no like eureka moment of me crying at the lab bench, being like, okay, this isn't for me anymore. It was much more of, okay, it was seeing specifically the postdoc I was working under, applying for grant after grant. And I think it was in his 5th year of his postdoc, and being like, oh gosh, I might have to do another postdoc because I'm not able to find a tenure track job. And I wouldn't say there was no one moment, but seeing that gradually over time, I started to think, okay, there's got to be more out there. And of course, exactly what you say is you're told academia, industry, you don't really know what else is out there. And I think it was by chatting with more and more people, that's ultimately how I learned what I fell into first, which was patent law. I think other people I've talked to as well, there's not been a defining, like, I don't know, one day your experiment didn't work and that's it. I think because we're sheltered, for lack of a better word, from other opportunities out there, until we know what other opportunities are out there, you don't know what else you could do with your life. And there's so many different job fits that might be right for you as well. And it might just be who you end up chatting with that day that gets you on a certain career path. So yeah, sorry, that was a long-winded answer to say I don't think there is one defining moment, but I think it's just a lot of, the scary part of networking and chatting with more people to figure it out.
If I could ask a follow-up question to that, when you decided to kind of leave academia, what kind of conversation did you have with your mentors, postdocs, like PI? How was their reaction? Were they at all supportive of you?
Yeah, that's a really good question. So mine was a little difficult to navigate, and so different people, different experiences. I was in a lab that they were very anti people leaving academia, to the point that was like my 2nd year of my PhD. I remember being sat down and told if I didn't want to stay in academia, I needed to find a new lab. So I was in please, I wouldn't say this is across the board. This is what I did myself. I just did it on my own, just down the down low, because you've got a lot of free time in your labs and when your advisors are watching over your shoulder. In an ideal world, you'd find an advisor who is supportive of at least you exploring non-academic pathways, because it will make your life a lot easier for internships, joining communities and clubs and all that. But ultimately, not everyone gets that sort of option. So I kept it kind of close to the belt. I told like PhD friends I was with, but I didn't tell my academic mentors. And to be frank, I wouldn't say that being it was a bit of a disservice to myself. Obviously, I did it for reasons so I didn't get kicked out of my lab. But I will say other advisors that I had were very supportive of me leaving academia. And I didn't find out till after I left. They're like, oh, actually, I had this connection with so-and-so here. That would have been a really cool person for you to chat with. So it would be advantageous if you're able to chat with your advisors, but it's tricky, especially in academia. The old guard don't like the idea of people leaving academia, even when there's less and less jobs available. Yeah.
So there's so much to choose from occupation-wise outside of industry. Like I could see myself doing just about literally everything. How did you decide or narrow down? Because you said you found groups and were talking to people. How did you narrow down from the million of occupations you could have chose from to first working in patent law?
Yeah. So one same. I still feel that same way when I hear a new job. Like, oh, that sounds super cool. I could do it. And I think that's part of the fun as well, is that I think a lot of us would be good in a lot of different jobs. So back in grad school, I think the thing that helped me most, say, land on patent law was it went down to chatting with so many different people, because I got to get a good look at, because even within patent law, there's, okay, all these different law firms you could pick with, and finding the right fit within a certain law firm, because each law firm's gonna have different cultures. So I think it's a lot about, because there's no one right fit, it's about finding the fit that works for you in the time that you're in. I hate, I sound like a broken record, but I keep just going back to it's the people you're chatting with. So going with the communities, especially like online Slack groups and things like that. And you'll pick these up for better or worse as you start chatting with people. Like I didn't find out about this science policy group I joined through Slack until I think it was like my 15th person I chatted with in science policy. I'm like, oh, hey, are you part of this yet? So yeah, it's a lot through making connections, unfortunately. And to be frank, I think the best way to learn as well is by trying something and figuring out if it's not for you. We've got a lot of, I feel like a lot of scientists were very used to, we need to be doing everything with the idea of the next 10 steps ahead. But there's a lot of transferable skills. Even when you say, I went to patent law for a couple of years and then I left it, I now know how to translate science to a whole new different audience, and how to read patents, and how to critically think in a whole new area. So I think if you're thinking about it and you're similar to me, where it's like, okay, all of this sounds really cool, I just pick one, go with it. And if you don't like it, there's always time to switch. And by having that network built up, you can always, it should be easier to start switching as well.
Thank you.
Yeah, sorry. I feel like I don't have a very good
No,
answer to any of these.
I think if there was one direct solid answer, then maybe everybody would be doing the job that they want to do or know exactly what they want to do post grad. But thank you for that.
Yeah.
I have another question. We've talked a lot about resumes and finding the right fit, but we haven't talked a lot about the interview actual process and the scariness that goes along with that. Do you have any other advice on actually getting to the interview and what to say and what not to say, how to research the company, things along those lines?
Yeah, for sure. So this will depend a ton on the sort of career that you're looking at. And sorry, I'm keep going back to it. The people you're chatting with leading up will be really valuable resources when you get to the interview stage, because especially if you've got that relationship with them already, they will, if not directly tell you, you can reach out to them and be like, hey, I'm really excited for this. Can you help me? And then they might even send you a list of questions to prep with. And that did happen to me in a few instances. Some are a bit more regimented. Like if you're interested in consulting, there's going to be like specific types of questions they'll be asking you about. And I would join in that case, like a consulting club or groups that will help you prepare for those interviews. And they are invaluable. So definitely check out some groups will even have like a interview section of their Slack channels. The other way I prepare for interviews is, if you can at all costs, ask them who you're meeting with, because it goes back to you're interviewing the person, not the company. If you can find out any sort of commonality or interest that you might share with one of your interviewers, it'll go from this big, scary, oh gosh, I'm chatting with, I don't know, some guy from Amazon, to, oh, I'm chatting with someone who also likes to bake like I do. And I would also, very importantly, make sure you have questions for them at the end. I've heard of people actually losing the job because it went super well, but then they ended with, do you have any questions? And they said, no questions for you. So even if you can't think of anything, try like look up the company, find something fun to ask them about. If you can't think of absolutely anything to ask the company, that goes back to interviewing the person. Ask them something about them. Maybe why they got into that career, why they picked the company that they're working with. Because I almost think of the interview as a two-way street. Because there are going to be a lot of companies out there that won't be a good fit for you, and you won't be able to tell that either until you're there, or at least maybe get an inkling of it during the interview process. Yeah, I also just thought of something else for Cole when you were talking about how to figure out from all the different things what might be interesting for you, is especially if you're a student, there's so many different internships opportunities to do, and kind of goes back to what you can get your advisor to sign off on. But like say for patent law, I found out that our technology transfer office had internships there. If you're interested in science policy, so many associations, a handful of associations have science policy fellowships you can do that supplement whatever you're doing in the lab, and postdocs, etc. So I tried checking out those because that's also an in-between before you take the full dive into a new career pivot.
Considered that. I hadn't considered that. Thank you.
Yeah. Oh, and you can also, it goes back to the network, but if you go back to them and say like, hey, I'm really interested in this, but I don't know if it's going to be a good career. How I made the technology internship thing happen was I actually chatted with them, and they said, oh, we don't have an internship program. And so I just kind of said, hey, I'm still interested though, and then they make it for you. So there's ways to make it happen, I feel like, but it all goes back to making sure that that they want to help you, I suppose. Yeah. Oh, I see another question in the chat. Kind of piggybacking off you mentioning leaving academia on the down low, how do you navigate job applications or interviews without a letter of rec from your PI? That's a fantastic question, because I was navigating that a lot myself. Find someone in your corner that is willing to vouch for you. I used my graduate program director who knew how my PI was, so I was able to kind of do that workaround. If you're get into any sort of graduate academic groups you're doing, like I was involved with our graduate student association, it can go a really long way if you can get a dean that's helping run the graduate student association write a letter of rec for you. Because you probably will. It looks the best if you have someone from your institution writing that letter of rec. But there's a lot of ways to get around. I'd say even if you're applying for academic jobs and your PI is not on the best, I would also go the finding it someone else, either another mentor or advisor in your corner. At the end of the day, by the way, you won't I wouldn't say completely lie to them. You will have to tell them if they're asking you what you're applying for and you're saying you're not applying for any postdocs. But there's a lot of that you can go with the gray or the in-between.
I think Anu had a question, but before we get there, I want to make sure you see this comment from Dr. Delgado. He says, thanks for sharing your experience. As a new PI myself, I think it really starts with us providing supportive environment for everyone, regardless of their future motivation. The problem I see on my side is that as PIs, we don't have very much guidance to assist students or postdocs in alternative directions.
That's a fantastic comment, because I also really strongly agree with that. That I've seen some universities are starting to work on programs for it where they're providing career panels, etc. But it is tough, especially as a new PI, figuring out how to, when you're trying to make your own name in academia, also trying to help your students figure out different careers is really challenging. So I think that goes back to the universities needing to provide a bit more support, and societies like PISA being willing to have more opportunities like this for students to get exposure to different careers. Ultimately, as long as you're supportive of them, though, I think that's all they can ask for, and open to them trying out different things.
Yeah. Anu, did you have a question you wanted to ask?
I think mine was more of like a comment. I just thought really exceptional talk and conversation, Chloe. I think it's very, very important. At my university, like I was shocked to begin grad school and see that there's so many career options that people can do. When I started in grad school is when I've seen someone who works on Wall Street on like as like a science advisor for like a law firm. I've seen someone do the patent thing like you are doing. I've seen someone work for TD Banks who like buys shares in like science-based, like companies. I've seen someone on the selling side. Like I've seen so many people doing so many like outstanding things. And I think like for me, it was just kind of a shock to see that there's all these things that you can do with your science degree, and you don't necessarily have to stay in academia or go to industry. So I just really wanted to like say that I think this is a great opportunity for everyone to hear that and just echo everything you're saying. Also, I had a comment about whether or not you should tell your PI your like true things. And I also like agree with you that maybe you shouldn't really do that. Whenever people come to like I recently had a student who had to leave his lab because his PI said like, oh, well, you're wasting my time if you're not going to stay in academia. So, and in that case, I was like, maybe you shouldn't really be so vocal about the fact that you want to leave. So I guess like my last question to you would be like specifically advice on that. Like, do you think it's right for, students to just kind of keep it on the down low, but maybe use their outside resources to prepare to make that jump? Or should they be so forward about just telling their PIs like, yes, I'm leaving academia and, stuff like that? But
I
great
think it's a
talk.
balance at the end of the day, and you'll be able to get a good read on who your advisor is and how they'll navigate it all. I mean, I will say I would never go into, even if you're starting your PhD and you're not going to stay in academia, if you go into any lab and say, hey, I want to start a PhD here, but I don't want to be here long term, you're going to rub people the wrong way, right? And so I also think closing off the door super early would also be the wrong call. So I think going and just saying you're open to different things is more the way to go about it. And then if you're having a very pushy PI, maybe closer, you deal with that conversation closer to the end. But I would try not to rule anything out right away. Yeah.
You mentioned having, or Christina, did you have a question?
Yes, thank you. This is Christine from Michigan State. Very excellent talk because I just recently defended my PhD, so it's really inspiring.
Congratulations.
Thank you to hear about all this and then consider the next step. I have a quick question. So, it's mainly, I guess it's more practical question. So when recent graduates or trainees that maybe in 1 or 2 year after postdoctoral training and then seeking to transition into industry, like what do they view those non-industry experience as experience, or do they only consider like the previous experience in industry as work experience?
That's a really good question because it's kind of, I'd say this is a gray area question. Different people have their opinions on different things. I am a huge advocate of counting even your PhD time as work experience. If they say that they want, I don't know, 2 to 4 years of experience, lab experience, or work experience, your PhD counts as that in my books because you are doing a very demanding job on top of classes in a lot of cases. So to answer your question, I would definitely count it. Now the question becomes if they start saying they want a specific, do you have industry experience in this? Or you can't lie and be like, well, I've got industry experience when I don't. But I'd work with that gray in between. If they say that they want an X number of years of working experience, for sure count your PhD and your postdoc years as that.
So do you think, in your opinion, what's the specific difference that's between the training in academia as postdoc versus training in industry as postdoc?
Also a great question because I mean, I've not worked in industry myself I did a summer internship. I don't really count that much. there isn't that much of a difference. I think it's kind of being nitpicky on the industry part. They want you to understand how the organizational structure work, which fundamentally that's going to be the big difference, how the organizations run in industry versus academia. But other than that, most of the techniques, the day-to-day is going to be pretty similar. And that'd be something to emphasize when you're looking at industry jobs. If they're asking for that certain number of industry years experience, or even put at the top as a header, like, under your name where you put like, I'm so-and-so, or I have like, I'm a biochemist at the intersection of XYZ. You could put with X number of years of research experience. So you're not saying industry experience, but you are still saying that you've got a lot of experience under your belt.
Thank you.
Yeah.
Does anyone else have any pressing questions for Chloe? Because I'll ask her a ton with our time we have left.
I have maybe a quick question. I was curious, you said that you've worked in so many different fields, not within the industry per se, but how difficult or how easy do you think it would be to switch career paths after graduating from a PhD and then going back to academia if you have an epiphany and say, you know what, I loved this. I want to go back.
That's a tough one, because the tough part, I'd say, is not about, leaving academia is going to be some sort of hurdle, learning curve, because how companies, how anything outside of academia works is quite different. It's doable though, because tons of people do it. The getting back into academia is actually interesting, because historically I've heard it's very frowned upon in academia to leave and then try to come back. Specifically, people do that a lot in industry, where they'll leave and then they might try to come back. But I have heard, and I've not tested this, but I have heard the past few years it's becoming increasingly more common for people to switch and come back into academia. I have heard this more from the industry perspective of, say, they worked in, I don't know, a research lab for Pfizer for a few years, didn't float their boat, so they were able to get hired back as a postdoc or a staff scientist in academia. I will say, though, the people that usually leave academia, at least they're not going to come back to academia for a bench science job. I do know quite a few people say they'll leave academia to do medical writing or some sort of science journalism and things like that, and then go back to the academic settings, but not to be a bench researcher, but instead to help a lab or a department or group write academic papers, write their science communication, etc. And that blends both fields really well together. So it's not impossible. I just say it's kind of, you got to work the angles right, I suppose. It's becoming increasingly more common, though.
All right, thank you.
Yeah.
We have about 5 minutes left. I think I want to go back to, if I loved the first beginning slide that you have about the definition of a scientist, and that it's not maybe exactly what we think. And sometimes we all have little mini crises. I have them plenty. So I was wondering if you could talk about what kept you motivated or sources of inspiration during those time periods of like really questioning or getting down in the toggle of networking and the endless hours that go into that, the people not responding to you, those.
Yeah, definitely. Because it definitely isn't easy. You've got to give yourself a pat on the back for even starting to begin with. For getting through, I think one, having your sense of community, your friends. Science friends, great, but I actually think having non-science friends is, if not equally, more valuable. My husband is not a scientist at all, and having him as a sounding board of when I feel like I'm going crazy, it is really valuable. So having that sounding board, the community, is really important. But also, I think something I did in grad school was I'd write down at the end of each month things that I was proud of doing. I used to do weekly and then it became too much, but every month write down. And yeah, it could be big things like coming to the PISA conference. But it could even be small things like I got my Western blot working, or maybe not small, depending on how long you've been trying to do that for. But having that list of, I reached out to 20 people this month. Having your own internal markers of how you're feeling like you're working towards an end goal is really valuable. I also think having a timeline, maybe this is the researcher in me, I love having any sort of timeline brought down by months chunks of what I should be getting done in each month. And that's kind of what I'll go back to make sure, okay, yeah, no one responded to me, but at least I'm following along these benchmarks of success. Because success at the end of the day is whatever you're calling it to be. Yeah, I'm trying to think if I've got any other ones. mean, you're not going to get everyone to respond to you, but I think if you come at it through the lens of you want to learn and that's what's being captured in that intro sentence or two, you're going to get, I'd say, quite a few responses. Yeah, I don't know if I have anything else off the top of my head, but I'll think on it more.
That was great. Well, if no one else has any pressing questions, we have 2 minutes left, but I really want to thank you for giving your time, giving an excellent talk, and motivating us. I'm leaving with way more encouragement. We love the cats. We already had a cat yesterday.
Oh no, he muted me now.
Oh, no, I can hear you.
Sorry.
Oh,
He's
you're
telling
good.
me he wants to go outside. Well, thank you so much for having me. It's been lovely. And if anyone has any other questions that they were, I don't know, too afraid to talk about here or wanted to chat about later, feel free to email me. I'll put it in the chat as well. But always happy to chat with people about this sort of stuff.
Yeah. So thank you so much. We really loved having you. And thank you to PISA for allowing us to have such a space to discuss this stuff. So yeah, I hope you have a great rest of your day. Feel free to listen to some science talks if you want, but if other than that, we thank you and wish you the best.
Awesome. Thanks so much. Lovely to meet everyone.
Okay. So I will hand it over to our next session moderators for liver metabolism and industry, which will be moderated by Taylor Petrucci and Anu.
Hey everyone, it's great to have you here today. Welcome to session 10, and thank you again to Dr. Kirk for that wonderful talk. I think I learned a lot about the things I maybe should have started earlier in my PhD, but it's not too late to start networking and making some connections. So if you get some LinkedIn connections from me later today, everyone in this talk, I have screenshotted your names. So I am excited to learn about liver metabolism and injury today. Myself and Anu will be here to keep the session going. If we have any time constraints, we will open up to questions after each talk, but they can be directed to the chat if we are running out of time and need to keep things moving. But without further ado, I'd like to introduce our first speaker, Alexandra Tomasovic from the Medical University of South Carolina, who will be presenting CYP2E1, how CYP2E1 mediates glycosylase 1 expression. So, Al, you can take it away.
Oh, can you hear me? Sorry, I forgot to unmute first.
Yep, you're good.
Perfect. So, yes. So my talk is going to be about how CYP2E1 mediates glyoxylase 1 expression. And I'm going to start by just giving you a little bit of background on what that means. So cytochrome P450 isoform 2E1, or CYP2E1, is this enzyme I'm going to be focusing on. It's a monooxygenase relevant to xenobiotic metabolism and quite a few different liver pathologies, including both alcoholic and non-alcoholic liver disease. And it's probably best known within the liver field for acetaminophen toxicity as it metabolizes acetaminophen. So CYP2E1 is known to generate reactive oxygen species, as well as the reactive aldehyde methylglyoxal, which I'll be abbreviating MGO going forwards. So methylglyoxal causes metabolic dysfunction and produces advanced glycation end products, or AGEs, which are known to further promote production of ROS and contribute to cellular oxidative stress. So on the right here, I have a schematic just showing the general distribution of CYP2E1 throughout the body. So it's expressed across all tissues, but it is primarily thought of as a liver enzyme. So methylglyoxal and these advanced glycation end products are widely affecting the body systemically and have a whole host of negative impacts. So they've been widely associated with a lot of disorders, just generally associating with aging, as well as metabolic dysfunction, diabetes, and many other conditions. So as an overview of the methylglyoxyl pathway and glyoxylase, so this is the glyoxylase system. And you can see on here the route for how CYP2E1 is involved in the formation of methylglyoxyl. Again, that's the big MGO in the middle there. And according to the literature, most of methylglyoxyl is formed as a glycolysis byproduct. And these glycolysis byproducts undergo spontaneous dephosphorylation into methylglyoxyl. However, it can also be formed enzymatically through CYP2E1. And GLO1 is, which is shown on red there, is the primary route for how methylglyoxyl is detoxified within the body. So 90% of methylglyoxyl is detoxified through GLO1 into D-lactate downstream. So any failings within this glyoxylase system lead to an accumulation of methylglyoxyl and leads to this perfect storm where you have rapid formation of AGEs, ROS, and many different negative health impacts. So GLO1 is also the rate limiting step in this pathway and is necessary for successful metabolism of methylglyoxyl. So some preliminary data in our lab, this was actually an incidental finding through our CYP2E1 knockouts. And we had found that there were huge alterations in GLO1 within the brain. We followed this up with some qPCR, saw that as well there. So here you can see there's about a 50-fold or more reduction across different brain regions of GLO1 transcriptional levels. So the hypothesis from here was that CYP2E1 plays a regulatory role in methylglyoxyl metabolism, somehow through controlling the expression of GLO1 in a tissue-specific manner, and that this is going to affect the overall methylglyoxyl levels within the animal. So the methods we were using for this, we are using a CYP2E1 knockout strain, as well as wild-type mice in the 129 background. As I just showed, some of this was done with RNA-seq, looking at transcriptional levels of GLO1. And then we're also going to look at some liver metabolomics data to investigate the metabolic products formed by GLO1. So I've put that schematic back on the side here so you can see how GLO1 relates to the formation of lactoylglutathione. And you can see on our metabolomics data in the liver that there is a decrease in lactoylglutathione levels in the liver. So this is consistent with the GLO1 decrease that was seen on qPCR. Next, we're going to continue the story by looking at Western blotting to probe the level of expression across several different tissues, as just because this is seen on a transcriptional level doesn't necessarily mean it is on the protein scale. So I'm just going to quick show you some overviews of what our Western blots tended to look like. We have knockout male, wild-type males, knockout females, wild-type females being shown here. This is looking at kidney. We did this in a whole host of tissues. So I'm just going to show you 2 or 3 here, and then I'll give you the summary schematic as well. So as you can see, huge significant decrease in expression of GLO1 in the kidneys within our knockout animals. And this doesn't appear to be sex specific or anything like that. So this again was repeated in the heart where a similar trend is seen. And then we've also got some small intestine. This is specifically looking at the ileum where you still have fairly high CYP2E1 levels normally in our wild types. And again, we see a huge change between the wild type and knockouts for this. So here's our summary with the different types of tissue we looked at. And there's a significant decrease across all of these tissue types. So I kind of summarized the fold reduction over here, but it is quite a reduction in the protein expression in all of our knockout animals. And So we're going to probe into that a little bit more here. So altogether, this is suggesting that there is a systemic impact of this deletion on the glyoxylase system that goes well beyond just within the liver. So lastly, we're going to look using LC-MS techniques to look at methylglyoxal levels within the liver. So to do this, we are kind of moving from this paper I have referenced here, doing a 4-PDA extraction and forming this adduct that you see at 175 is an adduct we see with our methylglyoxal. And then we also used myancerin, which is a tricyclic, as our internal standard. And overall, so here is the summary slide of that. We have an N of 4 for this for each group here. And I'm hoping to continue this moving forward in other animal type, or just to increase our N in general, sorry, excuse me. So this was interesting. We were expecting to see no sex differences, as none of the previous GLOW1 results had shown any indication that we would see sex differences. However, this is a statistically significant difference between the wild type females and their knockout counterparts in methylglyoxal accumulation. And then it's also statistically significant between both knockouts, in that there is a male and female sex difference being shown. So next steps include repeating this experiment and increasing our N, and then also investigating and diving into the proteomic data to look for some sex differences that may further explain this result. So results, overall, we see a significant decrease, about 50%, in GLOW1 protein levels across different tissue types. And then our liver metabolomics showed a significant decrease in D-lactoyl glutathione levels. And further, the HPLC analysis shows some sex-dependent differences in methylglyoxal accumulation, specifically that the CYP2A1 knockout females have increased methylglyoxal levels, indicating impaired handling and detoxification of methylglyoxal in the females compared to their male knockout counterparts. So in conclusion, our data reveals that deletion of CYP2A1 results in a consistent and significant downregulation of GLOW1 across multiple organ systems. We've seen this not only in the liver, but also the heart, kidney, and small intestine. Further, this supports that CYP2A1 is involved in some sort of regulatory mechanism that sustains GLOW1 protein levels across physiological contexts, and indicates a previously unappreciated role for CYP2A1 in mitigating methylglyoxal toxicity, most likely through GLOW1 expression. So these results highlight a previously unappreciated function of CYP2A1, where it is not solely a generator of ROS, but also as a potential player in antioxidant defense through GLOW1 expression. Past literature has shown that CYP2A1 contributes to some basal reactive oxygen production, and that this ROS can activate NRF2, leading to an increase in antioxidant defense genes, including GLOW1, which is downstream of this. So our findings are consistent with this potential pathway, showing that in the absence of CYP2A1, this antioxidant defense system may be impaired and leads to a decrease in downstream GLOW1 expression. And as such, the methylglyoxal accumulation. Future directions. So one of the first steps is to expand the methylglyoxal quantification to include other tissues to know whether this is a liver-specific effect or whether this is more widespread. More work is needed to determine the mechanism that's driving these changes to the glyoxylase system and understanding the specific mechanistic relationship between CYP2E1 and glyoxylase. Further, we would like to investigate the relationship between CYP2E1 and acetone or acetal, as shown in that glyoxylase pathway. The relationship between CYP2E1 and methylglyoxal involves an intermediate step with acetone. So knowing how acetone is affected by CYP2E1 can give us a clue about how much of this is shuttling forward into methylglyoxal. And then we'd also like to dig deeper into the sex differences and see if there are some proteomic differences in autoketo metabolism that may explain how the males are handling and detoxifying methylglyoxal differently. So acknowledgements, I just wanted to thank my lab. I'm in the Hartman lab at MUSC. And in particular, Reagan Roberts, who is an undergraduate who has helped a lot with the methylglyoxal HPLC quantification. Further, our collaborators, Katie Chetta and Dan Sprague, who have also helped develop this methods. And Dan Sprague has helped us derive a standard for methylglyoxal that we've been using, and our 35 for funding. Thank you so much. Any questions?
We have time for some questions. There's one in the chat. She says, did you see differences in glutathione since MGO can scavenge glutathione?
So I haven't had the chance to look at glutathione yet. I know NAC in particular, and acetylcysteine is commonly proposed as an antioxidant that helps with a lot of CYP2E1 mediated liver damage. So there's definitely some relationship there between antioxidants and antioxidant scavenging and that, but I haven't specifically had a chance to look at the glutathione levels yet. Definitely on the to-do list.
I have a question if there's, actually I have two questions. So with your data, you were showing us sex-dependent differences, in how MGO was accumulating. Do you think that sex hormones can modulate, that CYP2E1 or maybe GLO1, or maybe it's something else happening downstream with those metabolites? And then my second question is, since you saw all those expression changes in, the different organs, do you think the effect is tissue specific, or is it something more systemic in how MGO is being detoxed?
Yeah. So I can actually, my next slide. So I actually just saw some of the sex differences this week. I hadn't actually run the male methylglyoxal levels until recently. So this is some of our proteomics, just trying to look at relevant genes here. And while we see a similar fold change in GLO1, this like HAGH is GLO2, and these are some different aldoketo reductases, aldehyde dehydrogenase, so some different glycolysis related genes and such in there, where even though we're seeing the same reduction in GLO1, some of the related genes and metabolic capacity might be a bit different across those two. Oh gosh, I clicked something, sorry. And then as far as whether this might be hormonally regulated, the literature doesn't report a ton of differences sex-wise. However, we have found within our own lab that there's differences in like liver regeneration, capacity, and such between our male and female CYP2E knockouts. So this definitely supports that. And there's some like autoimmune phenotypes we see more in the knockout females. And so these advanced glycation end products definitely could be related to the formation of these sex differences we're seeing on a more macro scale in our animals.
Okay, sounds good. Thank you very much. That was a very interesting talk. Now for our next speaker, her name is Prerna, and she will be talking about zone-specific functions of beta-catenin in maintenance of liver zonation. So take it away, Prerna.
All right. Good morning, everyone. My name is Prerna Checkingle, and I will be presenting our work titled Zone-Specific Functions of Beta-Catenin in Maintenance of Liver Zonation. This work was done under the mentorship of Dr. Manga and Dr. Kim at the Pittsburgh Liver Research Center. The liver is a vital organ responsible for several metabolic and detoxification processes. It is the largest.
Prerna, I'm so sorry to interrupt. You might want to have the PowerPoint. There you go. For some reason, I just couldn't see it like that. You're all good.
Okay. Wait.
It's
How?
just not in presenter view.
Yeah, you can just click play from the beginning up there, and then it should like go into presenter view.
Can you see it?
No, not yet. Yes.
Oh, what keeps happening?
Maybe you can try the presenter icon kind of at the bottom of your screen. And don't worry, we stopped your timer, so you're okay.
Wait, where's the presenter icon?
It looks like a little champagne glass at the bottom of your screen. Yeah, that one right next to the Zoom bar.
Can you see it?
Your notes showing. That's it. That's a better one.
That's it.
I'm not sure. One second.
Perna, can you just share the window instead of the whole desktop?
Yeah.
So unshare and share again.
Can you hear us?
Go to the presenter mode first and then present it and then start sharing the screen. So unshare the screen right now. Yeah. And then make your presentation presenting mode and then share that window instead of the whole desktop.
Is it sharing my slides still?
Perna, share your slides to me, to my email, and I'll present for you while you speak.
Okay.
Okay, while she figures out, sending her slide to her very patient and wonderful mentor, Kim, we'll just move on to the next speaker, and then we can have Brenna come back after that speaker. So next is Ridgway Case, and he'll be talking about beta-catenin / liver X receptor axis regulates cholesterol metabolism and inflammation in murine cholestasis. Ridgway, if you're here, can you share your screen and take it away?
Sure. Hello, is it working? My mic
Yeah, we can
connection
hear you.
is a
Yes.
little bit. Okay, perfect. Okay. Good afternoon, everybody. My name is Ridgway Case. I'm an undergraduate researcher at the University of Pittsburgh. I work in the NEJAC Bowen lab at the University of Pittsburgh, and I'll be presenting my work on beta-catenin and its effects on the liver X receptor axis and how that regulates cholesterol metabolism and inflammation in murine cholestasis. So a short background about cholestasis. Cholestatic liver diseases affect the bile ducts and the biliary tree, either slowing or halting bile flow and the efflux of bile. So this reduction or elimination of bile flow leads to bile acid buildup in both the liver and the bloodstream, and this can be toxic and clinically very bad. So clinically, this progresses through fibrosis, cirrhosis, and in very severe cases, liver failure. There are some treatments, such as in PSC or primary sclerosing cholangitis. There can be stents put in or dilations done, but in most severe cases, transplant is usually required. So our research is on how to slow the progression of disease and protect against this because transplant is not a great option, and we'd like to not do that. So our diet, or how we induce cholestasis, it's through something called LCA, or lithocholic acid. Lithocholic acid is a toxic bile acid that we feed our mice. So our diet, where lithocholic acid is in their regular feed, it inhibits bile acid receptor signaling and leads to apoptosis and necrosis, which eventually causes cholestasis. Prior work by our lab is very focused on beta-catenin and its effects on the pathobiology of cholestatic liver disease. So prior work by our lab has shown that suppression of beta-catenin, or complete knockout of it, activates FXR. And with activation of FXR, as seen in the graphic, it decreases total bile acid levels, and it alleviates hepatic injury through bile acid efflux. So as seen in this graphic, FXR stops the production of caspase 8, and once caspases are made, they progress to apoptosis, which is ultimately bad in this case. So we would like to activate FXR because that decreases the caspases and it stops the apoptotic pathway. So our hypothesis is that loss or knockout of beta-catenin will alleviate cholestatic injury caused by toxic bile accumulation. We knocked out beta-catenin by having it flocked. So we had two LOX-P site on either side of the expressed gene, and we had an albumin-Cre promoter that would knock it out. So specifically albumin-free, so that only beta-catenin was knocked out in the liver and not every cell in the mouse's body. So here are some of the data that we found. At baseline, we evaluated the both wild type and knockout at baseline, and then we fed them an LCA diet and evaluated them again. So we have our wild type and knockout at baseline, and as you can see, there's no difference. These are identical basically. But after the LCA diet, there is a very stark difference. In the wild type, it is much more inflamed, there's much more necrosis, and it is significantly worse than the LCA, or sorry, than the knockout after LCA. The same findings are shown here in pictures of raw liver. You can see in the knockout liver, written KO, you can see in the knockout liver that it is a single color, it is much smoother, there is no inflammation, it appears much healthier. And importantly, it is much smaller. You can see in the wild type liver after LCA, it has splotchy coloration, it appears inflamed, it's almost like bubbly, and that's an indication of necrosis, a lot of inflammation, and overall it's a pretty unhealthy liver. So we can measure the health, if you will, of a liver by the liver weight to the body weight ratio of the mice, and that is shown here in this graph at the bottom left, that in the wild type LCA, they have a significantly higher liver weight to body weight ratio than our knockout LCA group. Here, these three graphs are showing liver enzymes in serum, and they are all significantly decreased in our knockout LCA group compared to wild type. Again, all pointing to the knockout of beta-catenin in livers has a protective or alleviatory effect against cholestatic liver disease. We did RNA sequencing and ingenuity pathway analysis to determine the up or down regulation of multiple pathways in our mice. The most downregulated pathway out of anything was cholesterol biosynthesis, and that's shown here in this red box. That's important to know, and I'll talk about it more later, because bile acids are synthesized from cholesterol. So cholesterol being synthesized less provides less substrate for bile acids to be made. So in our knockout group, if they have less cholesterol, then that means that less bile acids can be produced. Here, in the orange would be upregulation and blue is downregulation. In this green box, we have our RXR, PXR, and CIR signaling pathways, which are all upregulated in our knockout group. And this again points to the effect that knocking out beta-catenin has against protecting against cholestasis. As shown in the schematic at the very start of the presentation, this is the pathway that I was talking about that protects against it, and this is all upregulated. Now here in this Venn diagram, we see genes that are altered in our knockout LCA compared to wild type. And all of these increased genes, of course, I'm not going to go through all of them, but an important one to point out is SALT1C2. This is a sulfotransferase, or basically it puts a sulfate group onto a hydrophobic toxic chemical to excrete it from the body. So LCA, lithocolic acid, what we were feeding the mice, is highly toxic and it's very hydrophobic. That's important to know, and I'll talk about that also more later. But hydrophobic bile acids are typically worse across the board. They cause more damage to cell membranes, they cause more damage to mitochondria, they make more reactive oxygen species, and they're much harder to excrete. The more hydrophilic a bile acid is, generally the opposite, and they're much easier to excrete. So SALT1C2, it puts a sulfate group onto the hydrophobic bile acids and makes it more possible and more easy for them to be excreted. So there's a lot of gene regulation going on. So these next two slides will be about genes both up and down regulated. Here, CYP7A1 and CYP8B1, you can see that after LCA, there's hardly any expression here. But even before LCA, at baseline, the knockout group had significantly less expression. This is because these are bile acid synthesis genes through the classical pathway, it's called, or the more normal way of forming bile acids. And it is beta-catenin regulated. So knockout of beta-catenin significantly limits the amount of bile acid that can be produced through the classical pathway. Now, after LCA, we have CYP27, which is significantly upregulated knockout, which is an alternative bile acid synthesis pathway. Here at the bottom left, we have bile acid importers. On the right, there are exporters. I'll go through this relatively quickly, but the important thing to take away is that after LCA diet, our knockout mice are importing less bile acids and they're exporting more, which both points to the same thing. Toxic bile acids are being brought into the liver less and being exported more, which is protective. So more gene regulation. After LCA, there was no significant differences in any of the nuclear receptors, which is what we thought would happen. And CYP2B10 and 3A11, which are also both detoxification genes, kind of like SALT1C2. SALT1C2 is a sulfotransferase. These are both hydroxylases. So instead of a sulfate group, these add hydroxyl groups or OHs, which also make the hydrophobic bile acids more water soluble and easier to excrete. We looked at the bile acid makeup in both the liver and the serum to see what the differences were. And while in the liver, they were pretty much identical and there was no differences, you can see that from these pie charts, there's a lot of differences. Now, I'm not going to go through all of them, but here we have our hydrophobicity index, and you can see that there's a significant difference in the average hydrophobicity between our knockout and wild type groups. So as I was saying earlier, hydrophobic bile acids are much more injurious. They're worse across the board. In our LCA diet, the wild type group was significantly had a significantly higher average hydrophobicity when compared to our knockout group. We did more RNA sequencing, and this is a lot of data, so I'll just give you the main points and what's important. On the left, in group A, we have we show that loss of beta-catenin downregulates inflammation. Now, this can be shown here through IL-33, which is an inflammation marker, and in our KO group, which is these orange columns, it is downregulated in almost all of them. On the right side, we have cholesterol synthesis, and in our KO groups, or knockout groups rather, cholesterol synthesis is repressed. So again, that points to the same thing as our RNA sequencing was shown earlier, in that cholesterol is being made less, so less bile acids can be produced, and there's less inflammation. Now we did IHC and flow cytometry. This is talking about inflammation IL-33, what was just shown in the RNA sequencing. In our IHC, we stained for IL-33, and as you can see here at the top, in our wild type group, there's much more staining. It's this dark brown, and in the knockout, there's significantly less. There's almost, it's much harder to see. In our flow cytometry, we're looking for ILC2 cells, which I'll talk about ILC2, ST2, and IL-33 in a second, but we can see that in our wild type, there is significantly more ILC2 than in the knockout. And then here, these counts, there's less ILC2 in knockout, and while not significant, it appears that there's a little bit less ST2 in our knockouts. So ST2 is a membrane receptor, and ILC2 sits on it. It's found on ST2, and ILC2 responds to IL-33 in the inflammatory response. So less of all three of these, there's less IL-33, there's less ILC2, and there's less ST2. That points towards less inflammation across the board and a much healthier liver in our knockout groups, also caused by, of course, the knockout of beta-catenin. So in summary, beta-catenin loss or the knockout of beta-catenin protects against LCA-induced cholestasis through the LXR-mediated regulation of bile acid metabolism. Inhibiting beta-catenin suppresses the inflammatory response as just shown by downregulating the LXR, IL-33, ST2, ILC2 axis. And when taken together, these data demonstrate a novel beta-catenin dependent mechanism for alleviating hepatic injury in patients with cholestasis. Now, I would like to thank Carrie Nejak Bowen, my PI and mentor. She was very helpful with this whole project. I would also like to give a special thanks to Pamela Cornett and Matthew Carson. Since I started the lab, they have both been so helpful with everything, and I seriously can't thank them enough. So thank you for listening, and if there's any questions, I'd be happy to answer them.
Thank you very much, Ridgway. That was exceptional. I think someone said it yesterday. It's always amazing when we see people at undergraduate level give such exceptional talk, and you've clearly done a lot of work. Since we're falling behind due to technical difficulties, if you have any questions for Ridgway's amazing talk, please put it in the chat and then he can answer you there. And if Prerna is ready, we can move back to her talk. If not, we can move to the next speaker.
Thank you, Anu.
I think if you click swap displays, it will move to presenter's mode.
Okay. Good afternoon, everyone. My name is Prerna Checkingle, and I will be presenting our study titled Zone-Specific Functions of Beta-Catenin in Maintenance of Liver Zonation. This work was done under the mentorship of Dr. Manga and Dr. Kim at the Pittsburgh Liver Research Center. The liver is a vital organ responsible for several metabolic and detoxification processes. It is the largest internal organ by mass and plays key roles in carbohydrate, protein, and lipid metabolism. For instance, it regulates glycogenesis and gluconeogenesis, synthesizes plasma proteins like albumin and coagulation factors, produces triglycerides and cholesterol, and metabolizes xenobiotics. In addition, of course, we know that the liver produces bile and controls its excretion, which is essential for fat digestion. The liver lobule is organized along the portocentral axis from the portal triad to the central vein, and the hepatocytes are divided into 3 functional zones: zone 1 near the portal vein, zone 2 near the middle, and zone 3 near the central vein. These zones differ in oxygen, nutrient, and hormone availability, leading to metabolic specialization of hepatocytes along this gradient. And this spatial organization allows the liver to efficiently coordinate diverse processes such as energy metabolism, detoxification, and biosynthesis. The Wnt zonation is not random. It's actively maintained by signaling gradients, and one of them is the Wnt and beta-catenin pathway, which is what we looked at in this study. Zone 1, the periportal region, is rich in oxygen and supports processes like gluconeogenesis, urea synthesis, beta oxidation of fatty acids, and cholesterol synthesis. And Zone 3, the pericentral region, operates in low oxygen environments and specializes in processes that require low oxygen. So that would be glycolysis, lipogenesis, bile acid synthesis, and xenobiotic metabolism. So let's take a closer look at the Wnt mechanism. When Wnt ligands are absent, beta-catenin is targeted for degradation by a destruction complex containing the APC, axin, and the GSK3 beta, and this is the Wnt off state. In contrast, in the Wnt on state, when Wnt ligands bind to their receptors, the destruction complex is inhibited, and this allows beta-catenin to accumulate, translocate into the nucleus, and activate the transcription of the Wnt target genes. And through this mechanism, beta-catenin helps maintain metabolic identity in pericentral hepatocytes. So our lab previously showed in 2022 in the Hu et al. Study that endothelial cells in zone 3 secrete Wnt2 and Wnt9b ligands. And these ligands activate beta-catenin signaling in neighboring hepatocytes, inducing expression of zone 3 markers like GS and CYP2E1. When Wnt2 and Wnt9b are deleted in endothelial cells, or when beta-catenin itself is deleted in these hepatocytes, zone 3 markers are lost. And this basically demonstrates that endothelial Wnt ligands are essential for beta-catenin dependent zonation. So this led us to our key question: what happens when beta-catenin is specifically deleted from zone 1, zone 2, or zone 3 hepatocytes? And to answer this question, we set up our experimental design, which basically, to dissect beta-catenin zone-specific functions, we deleted beta-catenin in different mouse models. CYP1A2 targets zone 3, IGF-BP2 targets zone 2, and GLS2 targets zone 1 hepatocytes. And we use tamoxifen injections to cause an induction-triggered Cre-mediated recombination, leading to beta-catenin deletion in specific zones. And then we went ahead and stained various zonation markers to determine the changes in liver zonation when beta-catenin was specifically deleted in different zones, which was never done before. So first, we confirmed that beta-catenin deletion did not have any overt structural damage. And we did this by looking at H&E stained sections from all 3 knockout models, which showed that liver architecture or morphology was not affected. And this indicates that acute beta-catenin deletion does not disrupt basic hepatic tissue injury. First, we examined the expression of GS and CYP2E1, which are classic zone 3 markers. When beta-catenin was deleted from zone 1 or zone 2 hepatocytes, the expression of zone 3 markers, GS and CYP2E1, did not change. While on the other hand, when beta-catenin was deleted from zone 3 hepatocytes, there was a loss of zone 3 marker expression around the zone 3 hepatocytes. And this confirms that beta-catenin is critical in maintaining zone 3 identity. Of note, there were some zone 3 cells that still express CYP2E1, which we suspect is related to the incomplete recombination, especially in hepatocytes near zone 2. Next, we examined the expression of zone 2 marker, IGF-BP2. Across all models, we observed minimal or no significant reduction in expression compared to the control mice. And this suggests that beta-catenin is not involved in the expression of IGF-BP2 or the expansion/reduction of zone 2 hepatocytes. And beta-catenin deleted zone 3 cells are not actively replaced by zone 2 hepatocytes as well. Cyclin D1. Cyclin D1 is an interesting marker because it's both a zone 2 marker and a direct beta-catenin target gene. And in control livers, cyclin D1 expression is mainly mid-zonal. But when beta-catenin was deleted in zone 1 hepatocytes, cyclin D1 expression did not appear to have significantly changed. However, when beta-catenin was deleted from zone 2 or zone 3 hepatocytes, there was a reduction in the number of cells expressing cyclin D1. And these findings basically suggest that beta-catenin's regulation of cyclin D1 may be zone dependent, in that expression of cyclin D1 in zone 2 or zone 3 cells are under the regulation of beta-catenin, whereas cyclin D1 expression in zone 1 is not. Finally, we looked at CYP2F2, a zone 1 marker. When beta-catenin is deleted in zone 1 or zone 2, CYP2F2 expression is not significantly changed in GLS2 and IGF-BP2 mice, suggesting that CYP2F2 expression does not require beta-catenin. More interestingly, beta-catenin deleted zone 3 hepatocytes acquired that CYP2F2 expression, indicating that the absence of beta-catenin is required for the CYP2F2 expression in zone 1. And this suggests that beta-catenin is required for zone 3 hepatocytes, where it is not for zone 1 hepatocytes. Beta-catenin deleted zone 3 hepatocytes acquire that zone 1 marker, further suggesting periportalization of these hepatocytes. So in summary, beta-catenin is a central regulator of hepatocyte zonation across the liver lobule. Zone 3 hepatocytes we saw had clear changes in the zonal marker expression. There was a loss of GS and CYP2E1, and there was an acquisition of CYP2F2 as well. And we saw that the absence of beta-catenin is required for zone 1 hepatocytes to maintain its identity. And at baseline, beta-catenin regulates cyclin D1 expression in zone 2 hepatocytes. What are future directions with this? Well, we would want to identify mechanistic pathways by which beta-catenin uniquely regulates gene expression in each hepatic zone. We want to explore crosstalk between Wnt signaling and other zonation regulators like the oxygen gradient and other signaling pathways. And we would investigate whether beta-catenin deletion affects functional outcomes such as liver regeneration after partial hepectomy. Thank you so much.
Thank you so much. That was a wonderful talk. For the sake of time, we're going to move on to Matthew Carson's talk, and I'd like to encourage everyone to send their questions for Prerna in the chat. He's going to be speaking to us today about the loss of cholangiocyte beta-catenin supports angiogenesis and hepatocyte reprogramming through NF-kappa B mediated signaling during murine cholestasis. So take it away.
All right. Are we all good? Can we see my screen? Okay, perfect. All right, so thank you for the great intro. Again, my name is Matthew Carson. I am a postdoctoral scholar in the Nijek Bowen Lab at the University of Pittsburgh. I want to thank everyone for participating and coming to my talk today, where we're going to talk about the role of cholangiocyte beta-catenin in, oh, what happened here? Oh, there you go. The role of cholangiocyte beta-catenin during marine cholestasis. So as Ridgway previously stated, cholangiopathies or cholestatic liver diseases are of cholangiocyte origin, in which the bile ducts are either the buildup of toxic bile or impaired bile flow causes biliary damage. And primary sclerosing cholangitis is a specific cholestatic liver disease that we're interested in studying in our lab, where there's currently no effective medical therapies other than liver transplantation. And despite liver transplantation, roughly 25% of these patients have recurrent disease. So there's a current need to understand the mechanisms underlying cholestasis and finding viable therapeutic targets. So kind of going off everyone else's previous presentations with cholestasis in the Wnt beta-catenin pathway, Wnt beta-catenin has been involved in a lot of different liver pathologies and cholestatic liver diseases, and that is another main focus of our research group. So for the two models of cholestasis that I will be talking about today and focusing on for my project are the MDR2 knockout model and the DDC feeding model. So the MDR2 knockout model causes cholestasis through impairing phospholipid transportation that leads to the accumulation of toxic bile in the liver, while DDC feeding causes porphyrin plugs, which impairs bile flow that leads to biliary damage. And for the paper or for the talk today, I'll be talking about the MDR2 knockout model, but the findings are paralleled in the DDC model as well. So if anyone has any questions after this presentation, feel free to ask about that model too. So for our study rationale, prior work has shown that the beta-catenin pathway has a pleiotropic role in cholestatic liver diseases. Notably, in the MDR2 knockout model, exogenous deletion of hepatocyte beta-catenin can exacerbate liver injury, while following DDC diet, activation of beta-catenin can support hepatobiliary repair. And while a lot of these prior studies have focused on using the albumin-Curie beta-catenin model, which knocks out beta-catenin in hepatocytes and cholangiocytes, or hepatocyte beta-catenin specifically, very little is known on the role of beta-catenin in cholangiocyte signaling in the pathogenesis of cholestasis. So for our purpose of this study, we wanted to look at the role of cholangiocyte beta-catenin signaling in cholestatic liver disease pathogenesis, which we hypothesized that following the loss of beta-catenin should exacerbate cholestatic liver injury through NF-kappa B mediated mechanisms. So a quick overview of the timeline. So again, I'll be covering the MDR2 knockout model. We use an osteopotin Cre promoter to knock out beta-catenin specifically in cholangiocytes. And we treated mice with tamoxifen for 1 week. Then following a 2-week washout period, we euthanized our animals. So quickly to validate our knockout, we performed CK19 beta-catenin co-immunofluorescence studies. And we can see at baseline, our knockout is very effective in deleting cholangiocyte beta-catenin, as we see there's no co-localization of beta-catenin in our cholangiocytes. However, following in our MDR2 knockout model with cholangiocyte beta-catenin knocked out, we do see a repopulation of beta-catenin positive ductular cells, suggesting that beta-catenin might have a role in hepatocyte reprogramming, which I will talk about later in this presentation. And by H&E stainings, we didn't really observe any changes in fibrosis or inflammation. However, in our H&E stains, we saw thickening of hepatic arteries, suggesting that loss of cholangiocyte beta-catenin can have a role in hepatic vasculature remodeling. So going back to this whole hepatocyte reprogramming, during cholestatic liver disease, hepatocytes can acquire a cholangiocyte-like phenotype after biliary injury to replace these dying cholangiocytes. And this is kind of to help promote biliary repair and help fix any biliary damage that might be occurring. So we performed lineage tracing studies looking at SOX9, which is a cholangiocyte marker with EYFP. to look at the possibility of any repopulation of EYFP negative cells or SOX9 positive hepatocytes. So as we can see in our DKO animals, there is SOX9 positive hepatocytes presence as well as EYFP negative ductular cells, suggesting that there is hepatocyte reprogramming occurring in our DKO animals. And to validate this, we performed triple immunofluorescence for beta-catenin, SOX9, and HNF4 alpha, which is a hepatocyte marker, and found that there was a trending increase in DKO animals trending increase in SOX9 positive hepatocytes in our DKO animals. Further, we saw an increase in SOX9 positive beta-catenin positive ductular cells in our DKO animals versus our cholangiocyte knockouts at baseline, suggesting that there is a repopulation and hepatocyte reprogramming occurring following loss of cholangiocyte beta-catenin. So typically, cholestatic liver disease is characterized by inflammation, ductular reaction, and fibrosis around the periportal region. So we evaluated this by CD45, CK19, and serious red staining. However, in our DKO animals, we did not observe any changes in hepatobiliary inflammation, ductular response, or fibrosis, suggesting that we're only really seeing changes in this hepatocyte reprogramming that might help slow the progression or prevent further injury following loss of cholangiocyte beta-catenin. Now, appreciating that in our H&E images, we saw changes in hepatic vasculature, notably thickening of the hepatic arterial walls. We wanted to look at changes in our endothelial cells following cholangiocyte-specific knockout. So at baseline, notably what we saw was an increase in the sinusoid diameters, and we also observed this in our DKO animals versus our MDR2 knockout animals. We also saw an increase in CD31 positive vessels in our DKO animals versus our MDR2 knockouts, suggesting that loss of cholangiocyte beta-catenin can promote angiogenesis and endothelial cell proliferation. And these findings were paralleled by whole liver QRT-PCR, which we see DKO livers have an increased expression of CD31 versus all the other experimental groups. And next, we performed alpha-SMA IHC staining and found that basically in our cholangiocyte knockout animals versus our wild-type animals, we saw thickening of the hepatic arterial walls. And this was also observed in our DKO animals versus our MDR2 knockout animals. And then again, paralleling CD31 results, alpha-SMA mRNA expression was increased in our DKO livers versus our all the other groups. And together, these findings suggest that loss of cholangiocyte beta-catenin has a role in endothelial cell crosstalk and vasculature remodeling during cholestatic liver disease. So the next thing we wanted to assess is really how these cholangiocytes are talking to these endothelial cells to cause these changes in the vasculature. So we performed liver perfusions and isolated the cholangiocytes specifically and performed RNA sequencing to look at pathways that could be regulating this whole endothelial cholangiocyte crosstalk axis. And notably in our DKO cholangiocytes, both versus wild type and our MDR2 knockout cholangiocytes, we saw increases or an upregulation of pathways such as angiogenesis, vasculogenesis, and vascularization. And in these pathways, pro-angiogenic genes such as VEGF, PDGFB, NOS2, angiopotin, and CXCR4 are all upregulated in our DKO cholangiocytes versus our MDR2 knockouts, as well as our wild-type cholangiocytes. So to validate these findings in vitro, we first silenced beta-catenin in small cell cholangiocytes, or SMCCs, and evaluated the expression of pro-angiogenic factors. And we found that following silencing of beta-catenin, we saw an upregulation of VEGF as well as PDGFB. And to evaluate how silencing beta-catenin can promote endothelial cell proliferation, we silenced beta-catenin and took the conditioned media from these SMCCs and treated endothelial cells or transformed sinusoidal endothelial cells, or TSECs, with this conditioned media from the small cell cholangiocytes. And we found that this conditioned media promoted endothelial cell proliferation at a higher rate from our silenced beta-catenin cultures versus our control cultures, which together suggests that silencing or knocking down beta-catenin can promote endothelial cell proliferation. So next, we wanted to evaluate transcriptional regulators that might be involved in this pro-angiogenic phenotype that we're seeing. And of note, we saw that the NF-kappa B complex was upregulated in our DKO cholangiocytes versus our wild-type and MDR2 knockout cholangiocytes. And in this NF-kappa B network, we saw an upregulation of pro-angiogenic genes such as angiopotin and NOS2, suggesting that the NF-kappa B complex might be having a role in mediating this cholangiocyte endothelial cell axis. And why we're interested in this NF-kappa B complex is because prior work by the Najek-Bowen group and others has shown that the beta-catenin can interact with NF-kappa B to regulate its activity. So in the cytoplasm, beta-catenin can bind to NF-kappa B to sequester its nuclear co-localization and suppress its downstream activity. However, following loss of beta-catenin, NF-kappa B can freely localize to the nucleus and be activated and have any downstream signaling that might be occurring. So we validated this by co-immunofluorescence staining, and we found that CK19P65 co-expression was higher in our DKO cholangiocytes versus our MDR2 knockouts and our wild-type cholangiocytes, which demonstrates that loss of cholangiocyte beta-catenin can promote NF-kappa B activation. So in summary, loss of beta-catenin in cholangiocytes causes an activation of NF-kappa B, which supports the secretion of proangiogenic factors such as VEGF and PDGFB that lead to vascular remodeling and increases our endothelial cell proliferation and angiogenesis. Now, what we suspect and future work is really focusing on is how these endothelial cells might be promoting hepatocyte reprogramming. And we suspect that during proliferation and injury that these endothelial cells are secreting TGF-beta, which has been shown to promote hepatocyte reprogramming. And that's why we're seeing these mosaic bile ducts that are both beta-catenin negative as well as beta-catenin positive in spots. So in conclusion, beta-catenin signaling in cholangiocytes is a novel mediator of cell-to-cell communication, and that this loss induces a pro-angiogenic phenotype and can support hepatocyte reprogramming, both which may be involved in preventing accelerated liver injury following cholangiocyte beta-catenin loss. Now, I want to thank everyone who's been involved with this project, especially my mentor, Dr. Najek-Bowen, who's been very supportive throughout my postdoctoral career, as well as everyone as part of the Najek-Bowen lab group and our collaborators, and finally, all the funding that's been supporting this project. So thank you again, everyone. Now I'll take any questions in the chat if we have time or in person.
Thank you so much for your talk. That was really impressive work that you're doing, and I'm sure that we'll have some questions appear in the chat for you. We'll move on to Shadi Strata. I'm so sorry for my pronunciation. Imaging, and she'll be talking about imaging cytokine profiles, revealing hepatocyte-specific cytokines and their potential role in chronic liver disease. So moving on. Thank you so much, everyone. I think you might need to unmute yourself.
Can you hear me now?
Yes, perfect. Thank you.
Hi, my name is Shady, and I'm an undergraduate student working in the Combo Lab of the Tulane University School of Medicine. Today, I'll be presenting my work on the role of hepatocyte-specific cytokines in CLD, or chronic liver disease. So a little bit of background on hepatocytes and immune surveillance. Hepatocytes are principal parenchymal cells of the liver, performing critical functions for metabolic regulation, detoxification, bile production, and nutrient storage, synthesis and secretion of plasma proteins. Beyond these functions, hepatocytes actively participate in immune surveillance. This is so because they are positioned at the interface between gut and systemic circulation. They are also exposed to microbial products and dietary antigens. Due to this exposure, hepatocytes are sense and respond to immunological cues, contributing to the liver's tolerogenic and inflammatory responses. A little bit more background on cytokines and hepatic immunity. Cytokines are the central mediators of immune surveillance. They orchestrate communication between immune and non-immune cells. A cytokine storm is uncontrollable excessive release of these cytokines linked to systemic inflammation. It is well known that immune cells are a source of cytokines. However, it is unknown if non-immune cells, such as hepatocytes, can also contribute to this. And like I said earlier, hepatocytes are liver cells that are known for secreting albumin and transferrin. And today in this study, I will be talking about investigating the potential role of hepatocytes in directly synthesizing and secreting cytokines. So my question of the study is, do hepatocytes synthesize and secrete cytokines in normal and diseased liver? And as I expressed earlier, my rationale is that hepatocytes are continuously exposed to microbial products and dietary antigens via circulation from the intestines, while cytokines are well-established mediators of communication between immune and non-immune cells. The extent to which hepatocytes contribute to the synthesis of cytokines and thereby participate in immune surveillance is insufficiently understood. So my approach for this project was to curate a comprehensive list of currently known cytokines and subject them to a screening using the Human Protein Atlas database. This is a large-scale database that maps all of the proteins such as cytokines in the human body and systematically evaluate the expression of cytokines in hepatocytes. I then identified a subset of cytokines that are explicitly expressed in just healthy hepatocytes and validated their expression in both normal and diseased liver using a preclinical mouse model that induces chronic liver disease. So the significance of my project is that it can reframe hepatocytes not only as just passive targets of inflammation, but also active mediators capable of modulating immune response through cytokine secretion. Identification of these types of cytokines can also be early indicators of liver stress and provide a potential framework for the disease detection and its progression, especially in the transition from steatosis to inflammation and fibrosis. Understanding the specific cytokines involved can also inform the development of targeted therapies, including cytokine-specific immunomodulatory agents. However, despite these promising insights, the precise source and regulatory mechanisms governing these cytokine productions in hepatocytes remain unclear, emphasizing the need for further investigation. So again, back to my approach, I took a comprehensive list of cytokines and subjected them to the HPA database or Human Protein Atlas screening. They used immunohistochemistry imaging, which is a lab technique used to visualize specific protein localization in tissues. It's also important to note that the tissue samples used for this procedure were all from healthy human individuals. However, demographics such as age and gender were varied for each tissue, or for each tissue sample used. So to validate my HPA screening inaccuracy, I first screened for acute phase proteins. Acute phase proteins are proteins that in the blood that change in response to inflammation, and they are also known to be synthesized and secreted by hepatocytes. So as you can see, I have a list of the upregulated and downregulated acute phase proteins, and with the brown expression that you can see depicts the level of expression of the cytokine within the tissue. So as you can see in MBL2 and CRP, there is a lot of expression of that cytokine. However, this can vary. As you can see in RBP7 and RBP3, the expression is not as strong, but there is a little bit of that hint of brown that indicates the level of expression of cytokine in the hepatocyte tissue or in the liver tissue. So going back to my approach, I had 233 cytokines comprehensively listed. They went through HPA protein screening, and I had around 133 of those cytokines present, with 75 stemming from hepatocyte-specific, 33 from non-hepatocyte regions, and 25 coming from both regions. I also categorized these cytokines between their different families and functional groups. So I had interleukins, interferons, tumor necrosis factors, chemokines, which were also broken down depending on their cysteine residue attachments to CC chemokines, CXC chemokines, CX3C chemokines, growth factors, lymphokines, and TGF betas. Today, I will be only talking about my interleukin, interferon, and tumor necrosis factor families. However, I have data for the rest of the categories as well. So just to give an example of the evaluation between hepatocyte and non-hepatocytes, I'll be looking at my interleukin family. So as you can see in IL-25 and IL-36B, you can see the brown expression is relatively within the cell or the hepatocyte, indicating that the cytokine expression is coming from the hepatocytes. However, if you look at interleukin 18 and LIF, the brown expression is more in the white areas within the tissue sample, indicating that they are coming from non-hepatocyte regions. So what I did with this interleukin family is I partitioned them into hepatocyte regions, non-hepatocyte regions, and if they were present in both, you can see the symbol and the names for all representative cytokines within this certain family. Finally, I just focused on the hepatocyte-specific cytokine regions. So this is the interleukin family with a symbol, name, and the respective image for each. I did the same for the interferon family. There were only just two for this family, so the IFN alpha and the IFN beta. And then I did the same with the tumor necrosis factors family. Something that's also pretty interesting to note is that the level of expression of the cytokines were also localized within different tissues. So like, as you can see in the light sample, the brown staining is in certain regions of that tissue sample, which I found interesting. Also, something to note is that within the hepatocyte-specific tissues, the level of expression varies depending on the cytokine. Finally, in conclusion, hepatocytes can selectively express specific cytokines in normal human liver. Hepatocytes may significantly contribute to cytokine release, participating in inflammatory processes during chronic liver disease development. Some future directions with my study. So as of right now, I'm going through the process of validation experiments for cytokine detection, including immunofluorescent staining and Western blotting. I will then do qPCR for cytokine expression in primary liver cells. And down the line, I would want to compare results in normal and diseased livers in different stages of chronic liver disease progression. I want to acknowledge Dr. Kambu and the other members of my lab, Kamal Baral, Arissa Mercer, Leah Spade, Ramsey Roner, and Arya Chandrasekhar, and also the organizations ASIP, PISA, and CELT at Tulane University for supporting my work. Thank you.
Thank you very much, Shady. That was absolutely amazing and also great use of time. We're back on track. So if anyone has any questions for her, please feel free to ask them. We have a couple of minutes. Actually, there's one in the chat from Cole. He says, have you considered quantifying, i.e. halo analysis, your IHC stained tissues?
Am I allowed to speak or?
Yes, please.
I actually haven't. That's a pretty interesting idea, and I will take that into account with my study.
Are there any other questions for Shady? Okay, if anyone has any questions, she's still here, please feel free to leave them in the chat and she can respond to your question there. And we can just move on to the next speaker, who is Rithwik Agarwal, also from the Nijat Bowen lab, and we'll be talking about the role of beta-catenin and serotonin signaling axis in hepatocyte reprogramming during cholestatic liver disease. Rithwik, if you want to share your screen and take it away.
Are you able to see my screen?
Yes.
Great. Hello, my name is Ritwik Agarwal. I am an undergraduate researcher in the Najak Bowen lab. And today I will be speaking about the role of beta-catenin serotonin signaling axis, hepatocyte reprogramming during cholestasis. As my members of the lab have already mentioned, cholangiopathies are diseases that originate in the bile ducts. Specifically, we focus on primary sclerosing cholangitis, or PSC. And currently, there are no effective medical therapies to halt PSC disease progression. One option is to get a liver transplant. However, in 25% of patients, this disease will recur. As my colleagues have mentioned as well, the Wnt beta-catenin pathway is a pathway of focus for our lab. It's role has been indicated in several liver pathologies, including cholestasis, NASH, and various cancers. In cholestasis, there is significant damage to the biliary epithelium, and this can result in the death of cholangiocytes. As you can see here, there's damage to the cholangiocytes, and the hepatocytes that are present in the liver, therefore, then acquire cholangiocyte-like phenotype, which is known as cellular reprogramming, and this may contribute to biliary repair. Additionally, prior work done by the Najak Bowen lab has shown that beta-catenin also has a role in hepatocyte reprogramming. These are two papers published by my lab that explain this. My study, we're specifically interested in serotonin. Serotonin is a tryptophan metabolite. It is synthesized by the gut, brain, as well as cholangiocytes in the liver. Um, Serotonin has also been indicated to promote liver regeneration in previous studies, and uh, activation of a serotonin Wnt beta-catenin axis promotes hepatocellular carcinoma cell proliferation. Using this prior work and literature study, we hypothesized that beta-catenin signaling is coupled with serotonin signaling to promote hepatocyte reprogramming in cholestasis. In order to investigate this hypothesis, we first administered DDC in mice. DDC causes porphyrin plugs in the bile duct, which then induces cholestasis. After this DDC diet was administered, RNA was extracted from the mouse livers and QRT-PCR was conducted. AML12 hepatocytes were also used and they were stimulated by serotonin. The transgenic mice that we use in our study is known as S45D mice. Within these mice, the serine present on amino acid 45 of beta-catenin is mutated to aspartic acid. This results in a form of beta-catenin that is not degradable, and it allows for constitutive activation of beta-catenin. For mice that were administered the DDC diet, one type of mice we used was just given the DDC diet, and then we gave the S45D mice specifically the DDC diet as well. We measured levels of alkaline phosphate, also known as ALP, and bilirubin in these mice livers, and we found that these mice have S45D mice have less injury. Additionally, we looked at the bile flow of both mice and found that the S45D mice had increased bile flow as well. To investigate this further, we looked at the control as well as S45D mice under the microscope and and found that the A6 positive hepatocytes were more prominent in S45D, and it shows that overexpression of beta-catenin upregulates hepatocyte reprogramming. We also did RNA sequencing and found that the serotonin receptor signaling pathway is significantly upregulated in the A6 positive hepatocytes from the S45D mice. These results suggest that beta-catenin and serotonin signaling axis contribute to hepatocyte reprogramming and potentially can slow injury progress. Furthermore, then we did the QRT-PCR on the RNA of mice liver and found that the serotonin signaling receptors, HTR2A and HTR2B, as well as the serotonin synthesis enzyme TPH2, were upregulated in the mice that were given the DDC diet. Furthermore, we measured levels of SOX9 in the AML12 mouse hepatocytes, and we found that when stimulating these hepatocytes with serotonin, expression of SOX9, which is a cholangiocyte marker, is increased by 4.5-fold. Therefore, then we also looked at the role of beta-catenin in this and saw that when we overexpress beta-catenin in AML12 cells, it also led to the upregulation of the serotonin receptor HTR2B. From my study, we can conclude that first, in cholestatic liver disease, we have multiple injured cholangiocytes, and in these cholangiocytes are going to produce serotonin, increasing serotonin within the liver, and hepatocytes have receptors present for serotonin, as well as beta-catenin pathway being activated. This axis is going to promote reprogramming of hepatocytes to cholangiocytes, forming this mosaic bile duct. In the future, the beta-catenin serotonin signaling axis can be further studied as a potential therapeutic target for biliary repair in cholestasis. Using these results, my focus is on SSRIs, which are selective serotonin reuptake inhibitors. Children and adolescents are given SSRIs to treat depression and anxiety. And there are multiple clinical studies that have shown that a specific SSRI called sertraline has induced cholestasis in children. So, but however, the effects of SSRIs on cholestasis are unknown, as well as the prolonged exposure of SSRIs during childhood, whether it would impact the development and progression of cholestatic liver disease is unknown. Therefore, currently in my lab, we are working on on seeing the effect of SSRI treatment in adolescents on cholestatic liver disease progression. Therefore, we have 4 groups established. Two groups will start out with normal chow diet, and 2 groups start out with a DDC diet at 6 weeks of age. Then when the mice turn 9 weeks of age, we'll be giving one of the normal child groups SSRIs, as well as one of the DDC groups SSRIs. Specifically, in this case, we are giving sertraline, and we will be seeing the impact of this on the cholestasis in the mice. I would like to thank my mentors, Dr. Najak Bowen, as well as Matthew Carson, for all their support. And I would like to thank all of the lab members who have helped me throughout my project. And I would like to open up the floor to any questions.
That was amazing, Rithwik. The undergrads are on fire at PISA 2025. And it's just amazing to see. You have a comment from Cole in the chat that says, great talk. I had not considered that SSRIs treatment in adolescents might have an impact on cholestasis. Super cool work, and I agree. So we do have a couple of minutes if anyone has any questions for Rithwik. Okay, if no questions, I do have one. Oh, it seems like someone.
Yeah, no, I have a question for Rithwik just to get it started. So, with the SSRI treatment, do you think it will either improve cholestasis or do you think it might exacerbate injury in your model?
So that's a great question. Right now we are not sure what exactly will happen, but in uh, in the case that we do see improvement of injury, there are chances there is increased transdifferentiation of the hepatocytes to cholangiocytes. However, if we see that the injury does not improve, there may be a mechanism involving the increased serotonin that is unknown, and we would have to study that in further detail.
Perfect, thank you.
I do have one question, Ritwik.
Yeah.
Do you have any idea on the source of serotonin during cholestasis? Like, do you think it's like gut derived, or maybe from some of your hepatic or cholangiocytes, or some sort of population like that, or maybe one of those receptors that you showed us?
So that's a good question. I think that serotonin is being when in cholestasis, due to the injury, there might be um, a positive feedback mechanism working. No, I'm not really sure exactly where it is coming from, but that is something we could look deeper into and see if that can improve, if targeting that uh, pathway could improve injury progress.
Okay, thank you very much. If are there any other questions at all for Ritwik? If there are none, then we have come to the end of this session. I want to thank my co-moderator, Taylor, and I hope that you all learned something and we maybe convinced you to become a hepatomaniac. And with that, I will transfer the baton to Cole and Shanaz for the next session.
Good evening, everyone. Welcome to session 11, Cancer Molecular Pathways and Therapeutic Strategies in Diverse Cancer. My name is Shanaz Bano, and I'm joining you from the University of Pittsburgh. I'm delighted to be co-moderating this session with Chloe Haldick from the University of Oklahoma's Health Sciences Center. We are very excited to start this session with an outstanding lineup of talks. This session will explore diverse molecular mechanisms that drive cancer progression and therapeutic resistance across multiple tumor types. Each presentation will last approximately 13 minutes, followed by, considering the time strain, 2 to 3 minutes of focal questions. Please feel free to type your questions in the chat box during the talk, and we will address them after each presentation or at the end if time allows. Let's get started. So our first speaker is Harsh Nathan Dongre, University of Bergen, Norway, who will be presenting on fibroblast-mediated ECM remodeling and lymphanogenic shift in HPV-driven cancer cells. Welcome, Harsh. Please go ahead and share your screen.
Thank you, Shanaz, and also all the organizers of the meeting. Wonderful talks, and I'll go ahead and share my screen. I hope you can, not this one. I hope you can see my screen now. So just to add to that introduction, I'm also a research fellow at the Vascular Biology Program, Boston Children's Hospital, in the lab of Diane Willenberg. And today I'll talk about fibroblast-mediated ECM remodeling and lymphangiogenetic shift in the HPV-driven cancer cells. So I have mostly worked on mucosal squamous cell carcinomas, and these comprise of the head and neck region, especially the oral cavity and the oropharynx, as well as the larynx. And mostly you have heard of cervical cancer, cervix. Those are also mucosal squamous cell carcinomas. And recently there has been studies into vulva and vagina and other anogenital cancers. So if you look at the histology, all these cancers have a stepwise progression from precursors to invasive cancers, meaning that they start out from a normal mucosa to a dysplasia or dysplastic lesion, towards an invasive carcinomas. Presence of lymph node metastasis or the metastasis to the lymph node is a poor prognostic factor. And in the recent years, human papillomavirus infection is a risk factor. If you look at the statistics, the head and neck cancer is one of the most prevalent cancers. So approximately 1 in 25 cancer-related cases are related to head and neck, whereas if you look at the mortality rate, it's 1 in 20, meaning that 1 in 20 cancer-related deaths are attributed to head and neck cancers. If you look at the treatment options, it's mostly surgery and radio and chemotherapy, and that depends on the stage. If it's a localized tumor, it's mostly surgery followed by radio and chemo, whereas if it's a metastasis to either the regional lymph nodes or distant metastasis either to bone or other organs, it's immunotherapy. The 5-year survivals are ranging between 55 to 75 for different types of cancers, with head and neck being the poorest. As I mentioned, HPV associated cancers are on the rise, and the prevalence is around 40 to 50% for this type of cancers. If you look at the life cycle of HPV, it's interesting. And one of the questions is, why does it affect only mucosal squamous cell carcinomas? The answer is it's because of the stratification. So the HPV in its life cycle needs the stratification of the basal, suprabasal, and different layers of the stratified squamous epithelia to be able to release the virus and infect other basal cells in the epithelium. So they mainly work from the early genes E1 to E7, where the E6 and E7 are the most potent oncogenes. If you look at what they do is E6 blocks the degradation of P53 and E7 blocks the retinoblastoma protein, leading to outgrowth of deregulated cells. And these are the most common oncogenes that are associated with HPV, and I'll talk about them also later. If you look at the clinical correlation of HPV, it's quite evident that in HPV negative cancers at T1 or T2 stage, there is not much association with lymph node mats. However, if you look at the HPV positive, even at low tumor stage, there is a chance of lymph node mats. So, and this we have shown that there is a positive correlation between smaller tumors and lymph node metastasis. If you look at the histology, so this is a kind of a common histology of both HPV positive and negative mucosal squamous cell carcinomas, whereas you have a normal region, a tumor that is invading into the underlying matrix. And if you look closely, these are the tumor cells, but the tumor is not just the cancer cells. It also contains other microenvironment like the nerve cells, immune cells, fat cells, lymphatics and the blood vessels, and also fibroblast. We and others have shown that when the fibroblasts are associated with cancers, they can have either tumor restraining interactions, such as restraining the classical hallmarks of cancer, proliferation, invasion, metastasis, or leading to immunogenic cell death. Or they could also have tumor promoting effects where they increase proliferation, angiogenesis, etc. Based on this, we asked ourselves the question, do they play an active role in lymphangiogenosis in HPV positive cancers? In order to dissect that, we started collecting fresh tumor samples from the head and neck and vulva squamous cell carcinomas. We dissociated them and sorted them into cancer cells, stromal cells, and endothelial cells. Then we upregulated E6, E7 in both the oral cells and vulva squamous cell carcinomas. And today, I'll just focus on the vulva squamous cell carcinomas. So we did all the classical hallmarks of cancer. We saw increased proliferation, increased migration. But one experiment that I want to leave you with is the invasion. So as I said, the squamous cell carcinomas, they need a differentiated epithelium. So we engineered artificial mucosa wherein we populated the collagen or the underlying matrix with fibroblast and allowed the cancer cells to grow at air-liquid interface. So initially they look something like this, and we lift them at air-liquid interface, and finally they look something like this in a mucosa. And you can see that if you let them differentiate, you can see a polarity of the basal cells and the differentiated upper layer. And when we did that, you can see that when there was no fibroblast in the collagen matrix, the neither the wild type or the E67 cells invaded into the matrix. In the normal fibroblast, when it was in the stroma, you see there's a differentiation. However, only in the cancer-associated fibroblast with E67 that you see invasion. So then we saw increased proliferation, migration, and invasion, as I've shown here. Then we injected these cells into the mice, into the nude mice, and we see that the highest tumor volume was observed in E67. This work is still undergoing, and we are looking at the lymphatic density. In order to tease apart the molecular mechanism, what we did was we co-cultured either the fibroblast grown in the bottom and the cancer cells are grown on top in a transval assay, or vice versa. When we looked at the fibroblast or the calves in co-culture with wild type and compared with the E67, we saw two growth factors that were upregulated at gene level. We confirmed this with ELISA, wherein we see that HGF was upregulated with the in co-culture with E67. When we looked at the cancer cells, their other counterparts, we see that this HGF or maybe PDGFB upregulate VGFC only in the E67 cells and not in the wild type cells. This we validated using ELISA and Western blotting to show that also the intracellular levels of the VGFC was upregulated. Then we conducted a series of small experiments wherein we did provide exogenous HGF and we blocked it. And we see that not only the intracellular, but also at the VGFC released in conditioned media was upregulated. Then we looked at when we blocked the ERK pathway using SCH, we see that both the intracellular as well as the VGFC released in the conditioned medium was changed or was downregulated. So in short, what I've shown you up till now is that when the cancer-associated fibroblasts are co-cultured with E67, they release HGF. This HGF goes and binds to c-Met, its cognate receptor, phosphorylates it, and increases downstream signaling through ERK. But what is the transcription factor that governs VGFC upregulation? And in order to study that, we looked at gene RNA sequencing. So, and that in RNA sequencing, what we could see is that in the E67 co-cultured with cows, we saw upregulation of blood vessel morphogenesis, which was absent from the wild type co-cultured cells. And we saw genes, classical genes like VGF-A, VGF-C, FGF, which were upregulated in E67, but not in the wild type. And I want to focus or highlight on these three genes, neuropilin-2, VGF-C, and SIMA3F, which I'll talk about in the next slide. So, and this was also observed in the E67 cells treated with exogenous HGF. And then we validated this gene sequencing data at protein level, wherein we see that in E67 transfer cells, neuropilin-2 is upregulated, which is also the case when it's treated with exogenous HGF and in co-culture with VCAFs, which was quantified and looked at ELISA level. So as I mentioned, neuropilin-2 is an interesting gene, and it caught our eye because it's one of the genes associated with both angiogenesis and lymphangiogenesis. So neuropilin-2 acts as it's a transmembrane receptor, not a receptor tyrosine kinase, and it acts as a co-receptor. When it binds to VEGFR1 or 2, leads to angiogenesis, whereas when it binds to VEGFR3, leads to lymphangiogenesis. However, it can also bind to a SEMA3F, and in complex with plexin, leads to inhibition of both angiogenesis and angiogenesis. And in a way, it makes sense because if we go back to the data, we see upregulation of neuropilin-2, VEGF-C, and downregulation of SEMA3F in the E67 calf cells. So moving forward, there are reports that HGF can also, so neuropilin-2 can also be a co-receptor for HGF-mediated MET, which we confirmed in the endothelial cells when we upregulated neuropilin-2, and we see that the phosphorylation is increased of MET, whereas it does not happen in neuropilin-2 upregulated cells. So this we also looked into in our cancer cells, wherein we downregulated when we assigned neuropilin-2, and we see reduced phosphorylation of the MET receptor. To test the in vivo significance, we generated inducible neuropilin-2 knockout mouse, which were administered or which were governed by tamoxifen it was administered 5 days, and where we confirmed neuropilin-2 deletion. So, and then we performed the classical corneal micropocket assay, which was with the help of Amy. What we did was we implanted a HGF pellet in the eye. So in normal mice, this is the limbic vessels. They don't, so these are the normals. And when we implant the pellet here, you can see increased angiogenesis depicted by the magenta CD31 and increased lymphangiogenosis depicted by the green live one staining. So in normal cells or in normal cornea, you don't see any angiogenesis. In the controls, you see increased HGF mediated angiogenesis, whereas we knock out neuropilin-2 by administering 4-HT or tamoxifen, you see reduced or almost none compared to the controls. And this we quantified using percentage vessel area, number of lymphatic sprouts, and was significant. So I hope I was able to convince you that the calf secrete HGF when in co-cultures with fibroblast, and this signaling is mediated through, or this receptor complex is stabilized through neuropilin-2, and it downstream of it lies ERK. And I did not show you the data about the transcription factors, but it's still ongoing, and we are zoning in either ETS1 or KLF4 as the transcription factor that governs VGFC. Again, I don't have that much time to show you all the data, but what we have done is we have taken this conditioned medium, put on lymphatic endothelial cells, and we see that it's also HGF, it's also neuropilin-2 mediated. And with this, I would like to acknowledge my past mentor, Daniela Costia, and my current mentor, Diane. Both of them have been very supportive, and of course, my funding agencies. Thank you. And I'm happy to take the questions.
That was a great talk, Harsh. We are about out of time for your session, but there are some questions coming in the chat. If you would like to read those and answer those on your own time, I'll also post my own question for you as well in the chat as Rachel's talk is ongoing next. And so
Perfect.
our next
Thank you.
talk, like I said, will be by Rachel Edens Valentine from the University of South Carolina. She'll be giving us a talk titled Microbes, Methylation, and Motors, Acinetobacter's Role in Silencing RAB8 in Colorectal Cancer. And so, Rachel, if you would like to start, I'll turn it over to you.
Thank you. Okay, hopefully everybody can hear me and see my screen okay. Thank you, Cole, for that wonderful introduction. And I'm excited to talk to you all today about the underexplored role of RAB8A in colorectal cancer and the effect the microbe Acinetobacter may be having on that role. So just to set the scene, many of you are probably aware of the increasing incidence of colorectal cancer in the United States, especially among younger populations. Currently, colorectal cancer makes up 7.9% of all cancer cases in the US, and mortality continues to be a concern, with colorectal cancer being the second leading cause of cancer-related deaths and projected to cause almost 53,000 deaths in the US this year. Like any other cancer, different factors can increase a person's risk of developing colon cancer. Some of these include genetic mutations, family history, diet, and alterations in the microbiome. And of the genetic mutations known to increase cancer risk, alterations in myosin motors have been shown in the literature to increase a patient's risk of development and progression of colorectal cancer. So myosins are a superfamily of motor proteins made of two classes. The conventional class of myosins that people are generally more familiar with are comprised of myosin 2s, which help with muscle contraction and relaxation. And the unconventional class of myosins, however, are known to walk along the actin cytoskeleton in cells to help transport cargo. And myosin motors are found and function throughout organ systems of the body, including the lungs, the reproductive tract, the gastrointestinal tract, and the liver. And our lab is especially interested in the non-conventional myosin 5B, which is highly expressed in the gastrointestinal tract comprised of the small intestine and the colon. So myosin 5B has been shown to traffic cargo along the actin cytoskeleton from the Golgi apparatus to the apical membrane and epithelial cells of the gastrointestinal tract. And in order to carry the cargo that it needs to traffic, myosin 5B gets help from binding partners known as RABs. And the two most common Rabs that bind with myosin 5B are Rab11A and Rab8A. And these Rabs function by binding the myosin head to a vesicle within the cell that contains the important cargo to be trafficked. So some of the important cargo that myosin 5B has been well characterized to traffic are ion channels, including sodium hydrogen exchanger 3, sodium glucose cotransporter 1, and the bicarbonate chloride exchanger downregulated in adenoma. And together, these proteins facilitate ion absorption and help create a gradient that drives water absorption in the intestine. So when we look at normal human colon tissue, and this beautiful staining was actually done by Dr. Sarah Dooley when she was in our lab, we see myosin 5B protein shown here in green expressed at the apical membrane of the epithelial cells, and this expression co-localizes really well with the apical membrane marker gamma actin shown here in magenta. And in the overlay, we can see that that co-localization is really evident in white. However, when we look at colon cancer tissue, and here I'm showing a stage 2 tumor, we get an obvious depletion of the myosin 5B expression and a similar loss in gamma actin due to the depolarization of those tumor cells. Again, we see this depletion of myosin 5B in a stage 3 colon tumor. So now, given the depletion of myosin 5B that we see in colon cancer, some research has been done on the role of Rabs known to bind myosin 5B in colon cancer. And in 2019, the Gal lab out of Rutgers University showed that colorectal cancer patients with low Rab11A had poor survival, and that Rab11A deficiency in mice increased tumorigenicity. However, the role of Rab8A in colon cancer has yet to be explored and understood. So to start, we immunostained a colon cancer array for Rab8A to see if we saw that similar protein depletion to that of myosin 5B. And here in the normal colon, you can see Rab8A in green, expressed throughout the epithelium, that similarly to myosin 5B overlays nicely with gamma actin shown in magenta. And as expected, in stage 2 colon tumor tissue, we see a depletion in that Rab8A expression and in the gamma actin. And like myosin 5B, this depletion is recapitulated in the stage 3 tumor tissue as well. So given the alterations that we see in myosin 5B and Rab8A expression in human colon cancer tissue, we wanted to further confirm that this depletion occurs on a population level. So for this evaluation, we turned to the University of Alabama Cancer Data Analysis Portal, which allows us to evaluate publicly available RNA-seq datasets from the Cancer Genome Atlas. So we first looked at overall changes in gene expression in colon tumors compared to normal tissue. And we found that Rab8A and myosin 5B were significantly decreased in tumor tissue compared to normal. Now, we also looked at this gene expression across tumor stages, and we saw that Rab8A was decreased in colon cancer tumors of all stages, as was myosin 5B. So to further confirm the pervasiveness of this depletion in the patient population, we also looked at gene expression across patient phenotypes. And so when we look at patient sex, we found Rab8A and myosin 5B to be significantly decreased in both male and female patient tumors compared to normal. Looking at ethnicity, we found Rab8A and myosin 5B to be decreased across patient tumor groups as well. And finally, looking at colon cancer patients of all ages, we found that Rab8A and myosin 5B were decreased across age groups again of patients compared to normal. And finally, looking at weight, we found again Rab8A and myosin 5B to be decreased across patient weight classes compared to normal. Now, with confirmation that gene expression of Rab8A and myosin 5B is decreased in colon cancer patients regardless of phenotype, we also wanted to confirm changes in protein expression in the patient population. And the University of Alabama Cancer Data Analysis Portal allows us to run this analysis as well. So when we look at protein expression of Rab8A and myosin 5B in the patient population, again, we found both to be significantly decreased in colon tumors compared to normal tissue, which was a great confirmation of the staining that I just showed you. So far, we've discussed the important role of myosin 5B and Rab8A in trafficking cargo in the colonic epithelium. And we've also seen that in colon cancer, myosin 5B and Rab8A are depleted at both the protein and gene level. So knowing this, we wanted to go back to our cancer contribution factors to see if we could identify a driving force behind this change. And we chose to dive into alterations in the microbiome because we know microbial factors can interact with the epithelium, altering epigenetics, protein function, and expression, and impacting carcinogenesis. Now, colon cancer patients have been shown to be especially susceptible to pathogen expansion in the microbiome, and the most common of these pathogens that infect the colon in these patients include Fusobacterium nucleatum, Bacteroides fragilis, and toxin-producing Escherichia coli. And a new colonic pathogen that's gaining interest in the field is Acinetobacter baumannii. So A. baumannii is of special concern because of its high mutagenicity and resistance to antibiotics. So with all of this in mind, we utilized an incredible tool called the Bacteria and Cancer Database that allows us to look at the correlation between different bacteria at the genus level and alterations in gene expression in cancer patients. So we queried the database for each bacterial genus in colon adenocarcinoma. And interestingly, when we queried for Acinetobacter, we found that RAB8A, shown here in teal, was inversely correlated with the microbe, meaning that when colon cancer patients had decreased RAB8A expression, they had an increase in abundance of Acinetobacter. So to further explore this correlation, we grew up the pathogenic Acinetobacter strain A. baumannii 19606 by inoculating in brain heart infusion media and allowing growth overnight. So from this culture, we're able to use the live A. baumannii, heat-killed A. baumannii that was exposed to temperatures of 60 degrees Celsius for 30 minutes, and A. baumannii conditioned media that we obtain by pelleting the bacteria with centrifugation and stereofiltering that conditioned media. So using the heat-killed A. baumannii and conditioned media allow us to parse out if the effects of the A. baumannii are due to interactions with bacterial membranes or secreted metabolites in the media. So we took the colorectal cancer cell line HCT8 and treated overnight with each of our treatment groups and with a BHI control. So after treatment, we collected cells from each treatment group to look at changes in gene expression of RAB8A by qPCR. And when we look at the BHI treatment group, we didn't see a significant change in RAB8A expression, meaning our bacterial growth media alone does not affect RAB8A. When we look at our live A. baumannii, we see a significant decrease in RAB8A gene expression in our treatment groups compared to control. So A. baumannii is in fact influencing RAB8A expression in our cancer cells. Looking at the heat-killed A. baumannii, we did not see an alteration in RAB8A gene expression, meaning bacterial membranes of A. baumannii are not likely to be altering RAB8A. And finally, looking at our A. baumannii conditioned media, we see a significant decrease in RAB8A gene expression, meaning the secreted metabolites from A. baumannii may be influencing RAB8A. So in this talk, we've covered the role of myosin 5B and RAB8A in trafficking cargo in the healthy epithelium. And we've also seen that in colorectal cancer, myosin 5B and RAB8A are depleted in both protein and gene expression. And we've seen how Acinetobacter may be contributing to the decrease of RAB8A in colorectal cancer. In continuing this research, we are currently growing normal colonic epithelium organoids and colonic tumor-derived organoids that we will treat with the same A. baumannii conditions that we treated our cancer cells with. And in evaluating the effect of Acinetobacter on these organoids, we'll look at alterations in gene expression by qPCR and in protein expression by immunostaining of RAB8A and myosin 5B. So with that, I would like to thank the Amy Ingevic lab, the Mindy Ingevic lab, the Kristen Ingevic lab, all of my wonderful lab members and our funding sources. And thank you to ASIP for hosting this wonderful virtual conference. I'm happy to take any questions.
Hi, Rachel. I have a question for you. That was a great talk.
Yeah, thank you.
I study in part of my thesis exosomes and or extracellular vesicles and their functioning and traffic throughout the cell. Your talk mentioned RAB8, which is a small GDPase that's also involved in intracellular vesicle trafficking. How do you think reduction of this protein and intracellular vesicle trafficking protein affects cellular communication and or protein production in the cells?
Yeah, I mean, it's definitely negatively affecting the cells, just the overall decrease in protein transport kind of, hinders the cell and its ability to function normally. And it's hard to say whether or not the depletion in RAB is a cause or a symptom of the cell becoming cancerous. But yeah, it's definitely having an effect. I'm not sure if that fully answered your question, but it's a great point.
It does. I think it'd also be something very hard to just explain and or answer in a 30-second session. But
Yeah.
it does look like you have quite a few more questions in the chat. If you would
Okay.
like to read those and respond to those, my co-host is going to introduce our next speaker and we'll move on because for the sake of time, we'll go ahead and just continue.
Welcome, Daryl, who will be speaking on presenting on resensitizing therapy-resistant anoxy metastatic melanoma cells. Hi, Daryl. Go ahead, share your screen.
All right, I'm doing now. All right, can you see my screen? Okay, so hi, my name is Daryl. I'm presenting my talk on resensitizing therapy-resistant anoxic metastatic melanoma cells. So just a little bit of background information about melanoma and the anoxic environment. Skin cancer is the most common cancer in the United States, with metastatic melanoma being the most aggressive form of skin cancer. The 5-year survival rates currently are 99% and 35% for local and distant metastatic disease, respectively. In this study, we are specifically interested in that distant metastatic disease. Specifically, we are looking at metastatic growth in which it outpaces vascularization. This creates areas of severe hypoxia or anoxia in the tumor microenvironment. Hypoxia is when there's low oxygen availability, and anoxia is when there is no oxygen availability. We were looking at anoxia specifically. Previous studies have demonstrated that cells can express hypoxia-inducible factor 1 alpha, otherwise known as HIF1 alpha, as an adaptation mechanism to hypoxia and anoxia. We are also interested in the anoxic effects on the MAPK-ERK pathway, as this pathway is responsible for cellular growth and differentiation. Overexpression of ERK has been associated with inflammation and cancer progression. We are specifically interested in studying the final effector molecule, phospho-ERK, as our target molecule throughout this study. Currently, for metastatic melanoma, BRAF inhibitors are commonly used in the treatment. However, there has been resistance to BRAF inhibitors that are noted, and that resistance is associated with disease recurrence and poor survival. As phospho-ERK is the final effector molecule of this MAPK-ERK pathway, we use this because if the pathway is working, phospho-ERK would be present. If the pathway is not working, there would be less expression of phospho-ERK and decreased levels of that molecule. We are also curious what other molecules have an effect on this MAPK-ERK pathway, and one of them that stood out to us is DUSPs, or dual specificity phosphatases. DUSPs dephosphorylate signaling molecules, including phospho-ERK. They are up and down regulated depending on the specific disease process. In this case, we are specifically interested in DUSP6 as it has been shown in previous studies to be upregulated by HIF1 alpha, and in turn, it inhibits ERK. There's little data currently explaining DUSP6's specific contribution to tumorigenesis in melanoma. So our hypothesis in this study, we hypothesized that as metastatic melanoma cells adapt to these anoxic metastatic niches, they increase HIF1 alpha expression, causing DUSP6 levels to rise. This increase in DUSP6 will reduce the expression of phospho-ERK 1 and 2, a key component of the BRAF inhibitor signaling target, therefore causing BRAF inhibitor resistance in the anoxic melanoma cells. In order to investigate this, we used both Western blot and MTT assays. In this experiment for Western blot, we used protein lysates from the human melanoma cell line A375 when cultured in both normoxic and anoxic incubators. We then used the Western blot analysis to assess the expression levels of HIF1 alpha, phospho-ERK, and DUSP6. We additionally use Western blot analysis to assess levels of phospho-ERK when we use the DUSP6 inhibitor BCI. For MTT we also use MTT cell proliferation assays to compare the effects of the BRAF inhibitor dabrafenib and the DUSP6 inhibitor BCI on these A375 cells cultured in both normoxic and anoxic conditions. Additionally, we use the MTT cell proliferation assay on the combination treatment of a DUSP6 inhibitor BCI followed by treatment with the BRAF inhibitor dabrafenib. Our experiments were performed in triplicate, repeated 3 times, and we used t-test and one-way ANOVA statistics throughout. So time for our results. I have a histogram summary of densitometric analyses of Western blot results showing the expression of HIF1 alpha, phospho-ERK, and DUSP6 in the melanoma cell line A375, cultured in both normoxia, as seen in the red lines, and anoxia, as seen in the blue lines, at 24, 48, and 72 hours. The data demonstrates an increase in HIF1 alpha in anoxia, a decrease in phospho-ERK in anoxia, and an increase in DUSP6 in anoxia. Corresponding with those graphs, I have images of our Western blot findings. These findings are all at 48 hours in normoxia or anoxia, respectively. All right, next I have my MTT results of normoxic and anoxic melanoma cells. These are still the same cell line A375, treated with 0.5 micromolar of the BRAF inhibitor dabrafenib. Our data showed a significant reduction in cell proliferation in normoxic A375 cells, which we expected. However, the treatment had no significant effect on proliferation in anoxic A375 cells. This is presumably due to the previously described reduction in phospho-ERK in these anoxic A375 cells, as phospho-ERK is that downstream effector molecule that we're targeting throughout this study. And next, I have a representative Western blot image on the left and summarizing graph for that Western blot analysis showing phospho-ERK expression in BCI-treated anoxic A375 cells. BCI is that DUSP6 inhibitor that we used. This data demonstrates that the inhibition of DUSP6 was associated with a significant increase in phospho-ERK 1,2 expression in these anoxic melanoma cells. This is suggesting that they may be resensitized, which we investigated a step further. And next I have my MTT assay results from investigating these A375 cells with the combination treatment. So first we have our DMSO control cells of A375 cultured in anoxia for 24 hours. Then we have these anoxic cells treated with 2 micromolar of the DUSP6 inhibitor BCI for 24 hours. And lastly, the combinatory treatment of the DUSP6 inhibitor BCI with the BRAF inhibitor dabrafenib for 24 hours. There was a significant decrease in cell proliferation compared to the control. So in conclusion, the inhibition of DUSP6 in these anoxic melanoma cells increases sensitivity to this dabrafenib treatment. So in conclusion, melanoma cells increase expression of HIF1 alpha as they adapt to their anoxic conditions. In anoxia, these cells also express higher levels of DUSP6 and have reduced expression of phospho-ERK 1 and 2, causing resistance to dabrafenib treatment. Treatment with DUSP6 inhibitors can lead to re-expression of phospho-ERK 1 and 2 in anoxic melanoma cells. So therefore, combined treatment with DUSP6 inhibition and dabrafenib leads to decreased cell proliferation, suggesting that inhibition of DUSP6 increases sensitivity to anoxic melanoma cells to dabrafenib treatment. So future direction for this study, we plan on expanding this experiment to investigate other melanoma cell lines, as right now we have our complete data with A375. We have some preliminary data with other cell lines, but we are continuing the work there. For acknowledgements, I'd like to thank the Department of Pathology at Midwestern University, my research professor, Dr. Luigi Strizzi, the lab faculty, Gustavo Untiveros, and I'd like to thank the funding for these studies. Thank you so much for your attention.
That was a great talk, Daryl. I'm going to ask a quick question for you real quick before I turn it over to some of the questions in the chat. Why do you think that, forgive me if I'm pronouncing the drug wrong, difrafimib, if you'll pronounce that, reduced proliferation only in normoxic but not anoxic conditions in the metastatic cancer cell line you used?
Yeah, so dabrafenib, it's a BRAF inhibitor. It targets in that metastatic, or sorry, the MAPK-ERK pathway. In anoxic conditions, that pathway is functioning entirely, so that dabrafenib has a target that it can function with. In anoxia, our findings are suggesting that there's changes in the cell signaling and the pathway that's involved with the cell growth and proliferation. So we're seeing lower levels of phospho-ERK in those anoxic cells, indicating something in that MAPK-ERK pathway is not functioning correctly, leading to a resistance to dabrafenib treatment as it doesn't have its normal target there in anoxia.
That was a great answer. I think I have one more now since you answered that. Following up, have you guys tried to titrate oxygen levels to see where like the optimal normoxic or anoxic conditions are, like where the drug stops working and where it starts working?
We have not yet, but that is a really, really good idea, and that's definitely something that I'll consider with the future direction of this project.
Sweet. Okay. It looks like you have 3 questions in the chat. If you would like to respond to those, I guess in the chat, and we can continue moving on with our next presenter, if that sounds good to you.
That sounds great. Thank
Okay,
you.
our next and final presenter of PISA 2025 is Anna Pettyjohn from the University of South Carolina as well. She'll be presenting her work in which she is looking at the consequences of P2Y1 pyrogenic signaling in colorectal cancer. And so, Anna, if you would like to share your screen and take it away from here, I'll turn it over to you.
Hey, yeah. There we go. Okay, 1 second. Okay, thank you for that introduction. What?
Anna, right now it's showing presenter view.
Oh, stop. What did I do? Yeah. Okay. Is that good?
So now it's showing the notes under your slide.
Green table. Up top. Yep. Okay, is that better? Sorry, guys.
Yes, that works.
Okay. Thank you for that introduction. So, I'll just get right into it. So, as we all know, all cells need to communicate with each other to coordinate growth, repair, and defense. This is key for maintaining homeostasis, but can cause disease when communication is dysregulated. One of the major signaling pathways involved in this communication uses purines like ATP and ADP. These purines are important signals for cell-to-cell communication to maintain cell health. Purines activate 2 kinds of specialized receptors. First are the P2X receptors, which are ligand-gated channels. And we also have P2Y receptors, which are G protein coupled receptors. Collectively, these purinergic receptors are known as P2 receptors, and there are overall 15 subtypes. Now, looking at some of these purinergic receptor function in healthy gut tissue, we've previously shown that of the 15 P2 receptors, only P2I1, P2I2, and P2X4 are expressed within the intestinal epithelium. Using calcium indicators as a readout for receptor activation, we observed that P2I1 and P2I2 are functional within intestinal organoids, where increases in green, as seen in the middle and far right video, indicates receptor activation. In contrast to robust P2Y activation in organoids, when we looked at cancer-derived intestinal cell lines like TD4 cells, we observed little to no activation of any of the P2Y receptors. This led to the main question of, is purinergic signaling altered in colorectal cancer? So just a quick background, since I'm sure most people have seen this. Colorectal cancer is the second leading cause of cancer-related deaths in the US, affecting over 150,000 Americans annually. This year alone, 50,000 deaths are expected from colorectal cancer alone. And not only does colorectal cancer affect so many, it is also among the most expensive treatment regimes, representing a great healthcare burden. To go back to answering this question, first we turn to RNA sequencing from colorectal cancer patient tumors and normal cells from the Cancer Genome Atlas Program database. This provides information on what the normal cells and tumors express at the gene level. So based on this RNA sequencing from the Cancer Genome Atlas Program, we see that there is decrease in P2I1, P2I2, P2I13, and P2X3 in colorectal cancer tumors compared to normal healthy cells. Of interest to our lab, the receptors P2I1 and P2I2 both are present in healthy gut epithelium. This decrease may suggest that these two colorectal cancer receptors act differently than healthy cells. As we see, P2I1 shows the greatest decrease in expression in the tumor, so I'll be focusing on this receptor for the remainder of this talk. Next, we wanted to look at some of the demographics from this RNA sequencing data and saw that P2I1 is decreased in all ages of individuals with CRC. P2I1 is also decreased in both genders, and we also see that P2I1 is decreased in all ethnicities, including Caucasian, African American, and Asian. And finally, as you can see by this graph, we observe a significant decrease in expression of P2I1 receptor across all the stages of colorectal cancer from stage 1 to 4. Based on this information, we wanted to further understand whether decreased P2I1 expression was a trait in cancer cells in vitro, so to better dissect its potential role in cancer. Since the previous data is from bulk RNA sequencing, which means it is showing the expression of all the different cells, we wanted to look more into the P2I1 receptor expression on the epithelium, comparing healthy to colorectal cancer-derived organoids. These are a reductionist model to understand native tissue in the epithelium. So as you can see from this graph, the gene expression of P2I1 in colorectal cancer organoids is significantly decreased compared to healthy controls, which shows the same expression in the epithelium. So this led us to our next question of what effect would P2I1 expression have on cancer cells? For this, we took human TD4 parental cells with little to no P2I1 and knocked in P2I1 receptor using lentiviral transduction to have an overexpression model. First, we wanted to look at the differences in morphology and baseline fluorescence in these cells. As you can see from these images, the TD4 parental cells have this round morphology when they are confluent, while the P2I1 knock-in TD4 cells are more cobblestone-like and seem to stack on top of each other. We added a calcium indicator called GCAMP6S to these cells and looked at baseline fluorescence and noticed that the P2I1 knock-in cells over here on the right are generally brighter than the TD4 parental cells. So it is known in the field that both P2X and P2Y receptors can signal through intracellular calcium signaling. Since we have these cells with different expression of the P2Y1 receptor, we next wanted to observe if these were functional by looking at the cytoplasmic calcium levels. So because of this, we can utilize cytoplasmic calcium as a readout of receptor activation by adding this calcium sensor known as GCAMP6S again into our cells. And essentially, these calcium sensors allow us to look at the calcium levels, which glow green. Whenever a receptor is activated, we see an increase in this green, showing that there's an influx of calcium. To look at the function of these receptors, we use an agonist treatment where we put a plate of cells on a microscope and can add certain agonists or drugs to activate the P2 receptors in our cells and can measure the fluorescence intensity. Here's a video of TD4 parental cells expressing the calcium indicator in green before and following treatment with ATP gamma S. On the right side, as you can see, ATP gamma S causes an increase in green, indicating that there are some active P2 receptors. As we observe broad purinergic signaling here, we wanted to further dissect active P2Y receptors using select agonist. First, we have our TD4 parental cells, and as seen on the left is the vehicle control, and on the right, a P2Y1 agonist or activator is added. You can see that there is little to no receptor function in these cells, and here in the graph, we can see the calcium traces over time of the fluorescence when the agonist was added at time 0. We can also see that none of the other P2 receptors are functional in these cells. Next is our TD4 P2Y1 knock-in cells, where you can see this robust flash of green, showing us that the influx in calcium to conclude that there are functional P2Y1 receptors. As we can see in this calcium trace time course, there is the peak in fluorescence when the agonist is added, but the other P2 receptors are not functional in these cells. Now, looking at the maximum change in fluorescence for these two TD4 cell lines, you can see the significant increase when the P2Y1 agonist was added in this bottom graph here, indicating that these cells have functional P2Y1 receptors. Since we saw that these receptors are functional, we next wanted to ask the question, does P2Y1 impact cell migration? So we took our two TD4 cell lines and used a pipette tip to scratch the cells on the plate and create a wound, and then could see as the cells migrated to close the wound over 72 hours. Looking at the TD4 parental cells on the top here and P2Y1 knock-in TD4 cells on the bottom, we can see the initial wound, and then after 12 hours, the parental cells have almost already closed the wound, but by 24 hours, they have completely, while the knock-in cells barely migrate at all to close the wound even after 72 hours. Now you can see in this graph, looking at the wound closure percentage, the TD4 parentals are in green, where they migrate to close the wound by just 24 hours, while the P2Y1 knock-in cells do not. This can also be seen in this graph, where there's a significant decrease in wound closure percentage in the TD4 P2Y1 knock-in cells. Overall, we have seen from this that overexpression of P2Y1 delays cell migration. Now, in summary, in colorectal cancer tumors, organoids, and human cell lines, we see this downregulation of P2Y1. But when we reintroduce the P2Y1 receptor back into these cells, they exhibit slower cell migration and have differences in their growth and morphology. Leading us to our working model, where in the healthy colon, there is P2Y1, so there is normal growth and responses, but in cancer, with this decrease in P2Y1, we think there is more proliferation, migration, and less apoptosis, leading to this cancer phenotype. Future work will focus on driving apoptosis and further characterizing the role of these receptors in cancer. If we could find a therapeutic target to activate P2Y1, we believe this could lead to more cell death or other responses. In closing, I'd like to thank everyone for their attention and thank PISA for this opportunity to share my work today. I'd also like to acknowledge my mentor, Kristen Ingevic, as well as the members of my lab and our sister Ingevic labs and our funding sources. And with that, I'd be happy to answer any of your questions.
I think you're muted.
Yeah, I did. Thank you. Hi, Anna. I have a quick question. You found decreased P2Y1 expression in CRC regardless of patient characteristic. How robust was this trend across the TCGA data?
Sorry, can you repeat that? My volume was like down. Okay.
Okay.
Yeah.
You found decreased P2Y1 expression in CRC regardless of patient characteristic. How robust was this trend across the TCGA dataset?
Yeah, we found this pretty robust decrease for all those demographics for we also saw it in all like weight classes and then genders and cancer stages, as well as different types of colorectal cancer. So we did see this like robust decrease in P2Y1 for all those demographics.
Okay. I think we still have 2 more minutes to take a few more question answer, but I don't see much. And
Yes, Anna, there's another question in the chat. Is P2Y1 loss a driver of tumor progression or just a marker of dedifferentiation?
Okay. I think that's a great question. I think we're not sure yet, since we're still trying to characterize P2Y1 and its role in cancer. But so I think that we don't really know yet, but we're going to look more into it.
Have you considered possibly finding a cell line, a cancer cell line that has low levels of P2Y1 and then overexpressing it?
Yeah, so
That might
yeah, that's
answer
what we
that
have
question.
done. So most of these cancer cell lines do have like little to no P2Y1, and so we have made like overexpression models in a few cell lines such as H229, TD4, and KCO2s, and we've seen like the same differences in morphology and growth with these overexpression.
okay, I don't see any more questions. So thank you so much, Anna, for the excellent presentation. And now we will conclude the session 11. On behalf of my co-moderator, Kolai Haldik, I want to thank all our presenters for their outstanding talks, and to all of you for your engagement and thoughtful questions. This session highlighted how molecular mechanisms, signaling pathways, and tumor microenvironment interactions shape cancer progression and therapeutic response. Truly an exciting area of ongoing discovery. Thank you again for being part of this discussion and for contribution to a successful final day of the PISA Young Investigator meeting. As we wrap up this session and the scientific program for the day, please stay with us for the closing remarks and announcement of award recipients. And I will hand over to our next session to Dr. Sarah Dooley from the Medical University of South Carolina and Maria Antonia Zamru from Tufts University.
Yay, thank you so much. Those were amazing talks on cancer. And I was loving to see all the Ingevic presentations, as beautiful as ever, is the Ingevic way. So that brings us to the last day of PISA 2025. So claps to everyone. Congrats for making it through. I just wanted to start off by saying how grateful we are for everyone showing up, asking good questions, presenting good work. Meetings like this don't really work when the people don't show up and support each other, and I think that PISA has a great community for that. So before turning it over, I wanted to give a great thank you to my co-leader Maria and the whole planning group, Anu, Cole, and Anna. It has been a pleasure working with you for the past 6 months or so with our late night meetings of trying to figure out how to put all these abstracts together. I think we did a good job. And I wanted to say thanks for PISA for giving us the space to really build a trainee-focused program and meeting that's exceptional and is really not common in the field. I also wanted to make sure we gave a good acknowledgement and thank you to our invited speakers. We had Dr. Dogra taught us on day one about the role of DCLK1 in
ovarian cancer. On day 2, we learned from 2 amazing professionals in the field, Dr. Wakefield and Delgado. They talked about the importance of I cannot say that word. Grit is a better word, and how to juggle all the trials and tribulations that may come along in the ride for success in science. And today, if you got to attend, we learned a wealth of knowledge from Dr. Chloe Kirk, who taught us about the importance of networking, and that networking is not that scary, and all of the important things that go along in finding your fit in science, as well as crafting your resume and interview skills. So those are our invited speakers. I want to also say thank you to all of our amazing moderators. It wouldn't have happened without you guys for fielding questions and keeping us all on track. And a special shout out to Chelsea for all of our marketing and behind the scenes, Lisa for bringing a smile to all of our faces and bringing important emails to our inbox. And thank you to Dr. Coleman, who always makes sure these meetings happened and makes sure to support us as trainees. So I think that's all my thanks. And now I will turn it over to Maria to have a few quick words before she introduces our other ACIP officials who will be announcing the awards for PISA 2025.
Thank you, Sarah. And you did it very beautifully, so I don't even have to say that much. But I just wanted to echo what Sarah said. Thank you so much to Sarah for being co-chair. This has been such a fun ride, and she made this so much easier than if I had to do this even alone. So thank you very much. And a huge shout out to the rest of the committee, Anu, Anna, and Cole. Just absolutely fantastic. And it was so nice working with everyone, just made everything so easy and enjoyable. I also wanted to give a huge shout out to all of the speakers who did a fantastic job. So much great science was shared, and it was so nice to see progress from other meetings that we've seen in the past. So that's been just phenomenal. Huge shout out to the moderators for taking time just to moderate and have questions in the back of their mind, to have all of the speakers just like feel heard. So that's been fantastic. Thank you to all of the special seminars that we had, as Sarah mentioned. Those have been so eye-opening and enlightening for all of our training, and just give us like a glimpse of the different paths in the future careers that we might have. And also huge shout out to Chelsea for just like doing everything that she does, everything behind the scenes. Wouldn't be possible without you. Lisa for absolutely just being Lisa. You've just been, as always, amazing. And I love emailing you so much these past few months. And obviously, thank you to Bill, who's been just like a huge mentor and just keeping us on track and just like emotionally and like stable throughout all of this with all of your with all of your mentorship. So with that, I would love to introduce our closing remarks to our ACIP president and my mentor, Dr. Pilar Alcaide from the University of Miami Miller School of Medicine, as well as to our president-elect, Jonathan Homeister from the University of North Carolina, to present the awards and any extra data about the PISA meeting.
Great. I am going to start this. So let me share my screen here a little bit. All right. All right. So you heard my name. I'm John Homeister. I'm the current president-elect. And the current president, Pilar Alcaide, and I are just going to give some closing remarks here. I'd like to say a few things about the meeting and recognize, again, some of the people, the critical people that made the meeting happen and so wonderful. And then Pilar will follow up with the award presentations and some additional closing remarks. So Sarah and Maria did an awesome job recognizing all the people that made this meeting happen. And so this is a little redundant, but I would like to recognize them as well on behalf of the ASIP leadership. And so here is the organizing committee, Sarah, Maria, and the others, Anu, Cole, and Anna. So thank you very much to all of them. This meeting wouldn't happen without them. And so big shout out and thank you to the committee. So the meeting that you just attended was composed of 51 abstract-driven talks submitted by a number of different trainees, 11 postdoctoral fellows, 26 graduate or medical students, and 13 undergraduates. Some of those undergraduates were supported by a program that the ASIP has for getting undergraduates into research. We call that the Summer Research Opportunity and Pathology Program, or SROP. So we want to give a little shout out to those folks and thank the ASIP for supporting undergraduates and getting their research career started. There were 11 scientific sessions, 4 invited speakers, 3 special sessions, and a total of 143 registered attendees that made the meeting happen. So all of you will have your abstracts submitted and published in the American Journal of Pathology. So this is kind of the flagship journal of the ASIP. So it's a great thing you'll be able to put published abstract on your CV. And please consider publishing your work in the journal when it comes time to do so. You also heard a big shout out to the invited speakers. They're shown here: Sumitra, Evan, Brian, and Chloe. Thank you very much for your excellent presentations. I can tell from the number and types and quality questions that you got that your talks were very highly received and regarded. And then also thanks to the session moderators. Their names are not here, but their pictures. And of course, they did an awesome job keeping the meeting going and on time. And we appreciate all they've done to make the meeting successful. So I think with that, I am going to turn it over to, oh wait, I've got one more slide that I inserted here. So as was mentioned, this meeting wouldn't happen without the support of the ASIP staff. I left Chelsea off here, so let's mention her first, Chelsea Brochart, and all she did for promoting the meeting. And I think some of the technical aspects of actually running it the last couple of days. Lisa for as director of meetings and membership. You know, she's the one that really kind of put this all together behind the scenes. And then the critical support by the executive officer, Dr. Bill Coleman. So thank you to the staff. And with that, I'll turn it over to Dr. Al-Qaeda to